Hello, everybody. Do you hear me? If my audio is off? Okay. I'm happy to welcome everybody who is present now at our conference, all our participants, to the second day of the Life Science section. We want to thank all speakers of yesterday's session for excellent presentations and participants, and also to thank participants for interesting questions. Today we also plan to have a busy session. We'll have today 6 oral and 18 poster presentations. We have minor changes in the program. In poster session, we will not see 2 presentations because the speakers will not be able to attend the conference. I mean, number 14 and number 18, and also number 17 will be absent, if I'm not mistaken. Please remember that your oral presentation should not be longer than 15 minutes and 5 minutes for discussion. Also, you may use a chat box for questions. And again, I inform you that during the session, a comment photo will be taken. It will be taken after the last oral presentation, approximately at 9 a.m. by UTC, by Universal Time. And very important information for all participants, after this session at 2 p.m. by Universal Time, we kindly invite all participants to the all participants of the Life Science session to join the roundtable discussion on the topic development of possible cooperation in Antarctica in the area of life sciences. So let us start, and I give the floor to the chair of the Life Science session, Iryna Kozeretska. Iryna, you're welcome.
Thank you very much, Valeria. I'm happy to see many participants from different countries. Thank you very much to coming today too. And we can start.
Hello, dear colleagues.
Yeah, Vishnyakova, okay?
Oh, yes.
Karina?
So let me please to share my screen and start. Yes, okay, we can start. So I have some material for cetacean spatial distribution near the South Orkney Islands during Antarctic krill fishery in Australian summer 2020 and 2021. And it should be noted that since all Antarctic animals more or less depend on Antarctic krill, any significant changes in food webs may entail unpredictable changes for entire Antarctic marine ecosystem. And the higher trophic krill predators like baleen whales could be a perfect indicator of the ecosystem shifting. Therefore, the continuously monitoring of all the ecosystem components is very important in order to follow any changes, especially those caused by anthropogenic activities, to be able to quickly respond and take appropriate conservation measures to save the biodiversity of the Antarctic living resources. Considering the above, as well as the difficulties and the expensiveness of the data collection in such a faraway offshore region as Antarctic, it's very important to research Antarctic krill feeders, even using the opportunistic approach. And as such, this kind of opportunistic platform, we used the Ukrainian the Ukrainian trawler Moria Soдружество fished Antarctic krill. Cetacean observations from this platform of opportunity were conducted on board during the cruise from the end of December 2020 till March 2021. In total, there were 82 days of the observation from the ship. The fishing grounds were in the CRILL statistical areas 48.1, Bransfield Strait, Southern Shetland Islands, and the Antarctic Peninsula, and 48.2, South Orkney Islands. But in this part of our research, we focused only on the summer special distribution of Antarctic whales near the Southern Orkney Islands. Visual observations of marine mammals were conducted from the port and starboard sides of the captain's bridge that has height 17 meters by 2 observers. In total, there were 137.5 hours from each side of the vessel. 2 data sets were recorded, effort parameters like observation details, time, date, weather conditions, vessel speed, and course, and so on. And sighting parameters, geographical coordinates, species and number of animals in a group, distance from the vessel, behavioral specific details, etc. On this slide, you can see the vessel track line that we recorded when we were on observations. Here, the point is that despite it was opportunistic platform, not specified for the cetacean observations, the fishing crew often used the presence of whales to locate krill aggregations and follow them. As a result, over the observation period, 1,200 sightings from the starboard and 1,400 sightings from the port side were recorded. It corresponds to at least 3,500 individuals. Baleen whales belonging to 4 species: humpback whale, fin whale, southern minke whale, and southern right whale were observed, and also there were a few sightings of killer whales, and there were no significant differences in species composition between observations registered from different boats. So approximately 70% of sightings were humpback whales, and more than 25% were fin whales, and other cetacean species accounted for about 5%. Distance to the animals varied from few meters to few 10 kilometers, but however, direct interactions of whales with the vessel were never recorded. Here we have some estimation of group size for 2 dominant species, and these data were averaged for 2 data sets from different vessel sites. So observed fin whale group size varied from 1 to 30 individuals. On median value, 4 individuals in the group, and humpback whales were in groups of 1 and 80 till 80 individuals. On average, 2 individuals in the group. Often, it turned out to recognize mother-calf groups of humpbacks. And beside, there were several records of mixed groups of humpback whales and fin whales or minke whales. The special distribution of fin whale and humpback whale occurrences is shown on this slide. it can be seen that the whales were found mainly on the shelf near the Antarctic islands. Here, whales were feeding on the Antarctic krill swarms, which were also fished by krill fishing vessels, including the Ukrainian trawler Море Содружества. And here we have some hotspots for the Antarctic whales located on the dumps of the depths at the junction of the shelf. Probably in these locations are also formed the densest aggregations of Antarctic krill. For convenience of calculations on the temporal dynamics, the observation period was divided into 3 equal intervals, 23 days long, to describe dynamic in local occurrences. Registered groups per hour of humpback whales and fin whales. In the first cycle, that corresponds to the end of December and first half of January, the occurrence of baleen whales was the lowest. You can see that it's about 4 group per hour for fin whales and less than 1 group per hour for the humpback whales, for the fin whales, I'm sorry. The maximum occurrence falls on the second period that corresponds to the second part of January and first decade of February. And it was 10.5 group per hour for the humpback whales and 3.6 group per hour for the fin whales. And further, for the last period corresponding to the end of the Australian summer, occurrence of the fin whales remained at about the same level as previous period, and for the humpback whales, it was decreased. It is notable that such dynamics coincide with the dynamics of the krill catch. In the future, it's planned to carry out a more complete mathematical processing of the data obtained, as well as to analyze data from fishing statistics. Thank you for attention.
Thank you to you, Karina. Dear colleagues, any questions? Okay. Unfortunately, we haven't questions again. Is Ivanchikova here or not?
Yes, I'm here. Sorry.
Okay.
I'm here now. I'm ready to share my screen to go on.
Okay, welcome. We are ready to listen to you.
Do you see my screen now?
Yeah.
And presentation?
Yeah.
Okay.
It's okay. Welcome.
It's okay. We have, my topic is photoidentification study of humpback whales near the southern Orkney Islands during Australian summer 2020-2021. First of all, I'd like to say that photoidentification is a technique which is used to identify and track individuals of a wild animals study population over time. It relies on capturing photographs of distinctive characteristics such as skin patterns, scars of the animals, or other marks. In cetaceans, commonly used dorsal fins area and tail flukes. For humpback whales, we use tail flukes. It is important to say that photoidentification is generally used as an alternative to other methods. It is not invasive, and that's why we are using it to study humpbacks. Humpback whale study was held during began from December 2020 and finished in March 2021. It was conducted during Australian summer and in the waters around southern Orkney Islands. Opportunistic-based platform was used for opportunistic-based platform was used the Ukrainian KRILL fishing trawler Boris Družstvo. Photos were taken from the main deck of the vessel, which is 7 meters high, and from captain's bridge deck, which is 17 meters high. Photos were taken by two photographers. We use such cameras, which you see that Canon EOS 70D with 7300 millimeters lens and Canon EOS 7D Mark II with 400 millimeters focal distance lens. About the results, 236 individuals were identified and catalogued from 352 good and high quality photographs of humpback flukes. 43 individuals were recited during the investigations. It makes 18% of all meetings of individuals. 3 individuals we met for 3 times, and one, our records holder, you can see it on a photo, which we met in 4 times. Minimum interval between recitings we count like 1 day, because we have recitings during the day also, but we don't count it like recite. And the maximum interval being between recitings was 34 days. The highest number of recitings is due to trawling routes around the southern Orkney Islands, because krill fishing implies moving around krill aggregations from day to week. So some of groups we met a couple of times during the course. On the next slide, you can see our recordsmen, whom we met 4 times and 3 times during our investigation. All individuals were also primarily divided in 5 groups from percentage of black on their fluke, from 0 to 100%. And as a result, we see that we most commonly met individuals with totally white fluke, which were 82 individuals, which makes 34%. And on the second place were all blacks individuals, 63 individuals, which makes 27%. And with other percentages, 25, 50, 75, from 26 to 39 individuals. You can see it on the chart. Also, I'd like to say that we tries to make fin whales identifications, photo identifications of fin whales is making not by tail flukes, but dorsal fins, and there we see a good mark for fin whale. So thank you for your attention. Any questions?
Thank you, Yulia, for your presentation. We have questions in the chat. Can you see the chat? If not, I can read for you. Can you see? No?
No, I don't see now.
Julia, thank you very much for the nice talk. Only one methodological question. You need to see the whale's tail. Thus, if you cannot see the tail, are you not be able to identify the whale? Can you use any other parts of the whale body for identification, or the researchers can always see the tail? Second, okay, let us the first question, and after this I launch the second.
Okay, now I see chat. I see that question is from Tatiana. Thank you for your question. Of course, researchers not like see the tail every time, but we want to see as much tails as we the most quantity of tails. But there are some additional marks which can be used for identification of individuals. Sometimes even for humpbacks, their dorsal fin may have such mark that we can identify that exact that individual. But it should be marked really very good. It may be some scars from vessels maybe, or something like that. So such identification is possible in that cases.
Okay. The next question is, sorry. Wow. You can see? Okay.
Yes, now, are really the tails so unique to identify the specific whale personality? Maybe that the first question will be what we understand under the personality. We are talking about individuals, so we know that they are that exact animals. But about personality, it's more hard to to distinct it, I think.
Okay, I have one small question to you. What about DNA samples? Have you such possibility or not?
You see, we use the opportunistic platform, the KRILL vessel, and we have we don't have
zodiacs for this
equipment.
But you show on it seems to me the first slide zodiacs, and that is why it seems to me maybe for the first time only that you can such possibility a special. Okay, look attentively at the first slide of your presentation, and it seems to me you will realize what about I put this question to you. Thank you.
Okay, but we have some opportunistic samples from humpbacks which came in troll. So it's like opportunistic biopsy, as we called. But we have no equipment for that, and maybe we will extend our program for next season.
Yeah, it seems to me if this vessel used zodiacs, it's maybe possible sometimes to make biopsy in the same time, not only photography.
Yes, and they can could be even more productive than like from Vernadsky station, because we met as from reports of Karina. You may see that we met a lot of humpbacks, so it is like more likely to get more biopsies, really.
Okay, thank you. I wish you success and fruitful work in the future. Okay,
thank
you very much. And now we invited our Malaysian colleagues, because person from Ukraine are later. Okay, where is our Malaysian colleagues?
Hiudayu, can you join? Can you join now? Hiudayu?
Yeah.
Okay.
Thank you.
Now, thank you very much. We are happy to see you today and welcome you to presentation.
Assalamualaikum and greetings to all present. I am Hiudayu Syuhada binti Adnan, a graduated master student from Universiti Sains Malaysia. Previously, I was supervised by Dr. Faradina American, and today I will be presenting my master research project, but I'm not going to present the entire project, but we'll be focusing on the main part, which is on the strong and widespread cycloheximide resistance in the eukaryotic algae genus Stichococcus. Okay, let me just explain the title so that everyone here will have a better overview of this presentation. Cycloheximide is an antibiotic that is widely used to inhibit the growth of eukaryotic microbes, while Stichococcus is a genus of eukaryotic microalgae. So basically, this algae did not die in the presence of antibiotic that should kill it. So this project was initiated by the discovery of uni-algal cultures of Stichococcus in culture medium supplemented with cycloheximide. Next slide, please. Okay. Now I would like to take all of you to the journey of the serendipitous discovery. In 2016, we obtained soil sample from Sydney Island, Antarctica, contain mixed assemblages of different microalgae, and the initial aim was to isolate cyanobacteria from this consortium. So to do so, next slide please, we supplement the growth medium, which is the BG11, with 100 microgram per mil cycloheximide. So after about a month of incubation, I saw some green colonies growing on the plates, and from the grass green color, I suspected that they are not cyanobacteria. Next slide, please. So this is how they look like under the light microscope. So for microalgae taxonomies, we'll know that this is not cyanobacteria, because the grass green color really show that this is not cyanobacteria, because cyanobacteria is blue-green. So, and my assumption was right. They are actually uni-algal cultures, uni-algal colonies of unicellular eukaryotic algae, green algae. And similar microalgae were observed for the rest of the plate. Next slide, please. After checking with the identification key, those strains were identified as Stichococcus bacillus, which is a unicellular green algae. Since cycloheximide resistance in green algae never been reported anywhere else, so we really take precaution steps to make sure this finding is valid. So a preliminary experiment was conducted. Next slide, please. A preliminary experiment was conducted to confirm the discovery by comparing the growth of Stichococcus, chlorella, which is also unicellular green algae, and also Clepsomedium, a filamentous green algae. So we compare between different green algae. So the strain were cultured in BBM, bowl basal medium, with and without the addition of cycloheximide. And the growth was assessed visually by taking photos of the plate every week. So as you can see from the slide, growth in treatment plate, the one with 100 microgram per mil cycloheximide, can only be seen in Stichococcus, and no growth was seen in chlorella and Clepsomedium. Next slide, please. So after the preliminary experiment, we decided to test the cycloheximide sensitivity on Stichococcus strain collected from other regions, from the Arctic. Next slide, please. From the Arctic, temperate. The Arctic is the Alaska, temperate from Germany, the tropic from Malaysia, which I collected myself, and also another strain from Antarctica, an old Stichococcus strain collected by Dr. Paul Brody in 1970s. Okay, next slide, please. So these are the list of sample used. Number 1 to number 6 are all Stichococcus strain, while the last two is the Chlorella and Cocomyxa. Both are green, unicellular green algae, which also included in every experiment, just to confirm that our cycloheximide is working well. So next slide, please. So a more sophisticated series of experiments were conducted to test the sensitivity of all Stichococcus strain towards cycloheximide by determining the minimum inhibitory concentration, the MIC, the growth rate, and also the time taken for the resistant cell to grow in the presence of cycloheximide, which was determined by the lag time. So next slide, please. So moving on to the method, result, and also discussion. To test the effectiveness before any experiment were conducted, we conduct this experiment first, which is we test the cycloheximide effectiveness. So plates of BBM and also BBM supplemented with 100 microgram cycloheximide were left on the bench under the incubation setting, 24 degrees Celsius and 27 micromole photon meter per second, were left on the bench under the incubation condition for a month. And after a month only, we start inoculate Stichococcus and also a cooperative strain, which is the Cocomyxa. Next slide, please. So after the incubation period, image of the plates were taken and the results, all replicates were similar. Thus, the plates here represent the rest. So the resistance strain, which is the Stichococcus CCAP, this was the one that collected from Antarctica, the old sample from Dr. Paul Brody I mentioned before. The strain showed a visible growth in both types of media, while no growth of Cocomyxa can be observed in BBM supplemented with cycloheximide. So this test confirmed that under our incubation setting, the cycloheximide still working very well, because Cocomyxa cannot grow in the presence of cycloheximide. So cycloheximide can still kill the eukaryotic microalgae. So so for the main experiment, serial dilution of cycloheximide was prepared by two-fold dilution following the protocol of broth microdilution, resulting in the final concentration ranging from 0.5 microgram per ml up to 1000 microgram per ml. 3 replicates were prepared for each concentration, and the assay was carried out for 15 days using sterile transparent 96 well microplates. Microplates. A wheat oil culture was adjusted to 1000 cell per ml, and 100 microliter of volume was injected into 3 replicates well at each cycloheximide concentration, and also 3 replicates well of positive control BBM without cycloheximide. So plates were sealed and to prevent evaporation, and the plates was placed under the same incubation setting mentioned before. So the growth, next slide please. So the growth of cell population in each well was assessed by measuring the chlorophyll fluorescence using microplate reader. The sample were transferred first to a black microplate for a higher sensitivity during the measurement, and the cell density of positive control were also determined by counting the cell using a hemocytometer. For assessment on day 15, the final day of incubation, we used to determine the MIC, the minimum inhibitory concentration. For the growth rate analysis, the growth rate of each strain was first determined using a software called growth rate. So the program determined the exponential phase of each strain and then calculate the growth rate in minute. For the analysis, only high resistance strain were involved. Oh, sorry, because the slide I converted into PDF. So yeah, it's like that. So for this analysis, only highly resistant strain were involved, and the comparison was made between control culture and the BBM supplemented with 1000 microgram per ml, which is a very, very high cycloheximide concentration, which is the highest concentration in the study. And for the lag time, the comparison was made between control culture and the lowest and the highest cycloheximide concentration. So next slide, please. So in this slide, we before assessing the growth using microplate reader, we see the pattern, the growth pattern in each plate, and by naked eyes, and from there even using we call it visual MIC. All the Stichococcus strain, Antarctica 1, Antarctica 2, Antarctica 3, Arctic, Tropic, are all they can grow up until the 1000 microgram per ml cycloheximide. And for the temperate strain, Stichococcus strain, they cannot. They died at 62.5. While chlorella and Cocopmyza, they died at 31.3 and 15.6 microgram per ml respectively. So next slide, please. Okay, this histogram showed the cell density of tested strain after 2 weeks of incubation. The result supports the visual MIC where all Stichococcus strains successfully grown in all cycloheximide concentration except for the temperate strain, and also the comparative strain which had lower MIC than the temperate strain. There was no significant difference in the cell density between the different cycloheximide concentration and the control in the Antarctica 1 and Antarctica 3. There was no significant difference in the cell density between the different cycloheximide concentration. However, for the Antarctica 2, there's no significant difference in the cell density between the different subconcentration. However, the control culture of the strain was significantly higher than the control. This means that the presence of cycloheximide affects the growth but not the cycloheximide concentration. Okay. Next slide, please. This is the growth rate analysis. Next slide, please. Okay. Okay, for the growth rate analysis, there is no significant difference in the growth rate between the control and the treatment, although there is a great significant difference in the cell density between the control and treatment of strain Antarctica. Which Antarctica? Oh, sorry. There is, as you can see from the histogram, there's no significant difference in the growth of the cell in the control and treatment for Antarctica 2, Antarctica 3, and the Arctic. And as for the tropical strain, there is no significant difference also between the control and the treatment. Next slide, please. Okay, this is the lag time of one of our aim is to see the time taken for the resistant strain to start growing in the presence of cycloheximide. So we compare between the control, which is no cycloheximide added, and the lowest cycloheximide concentration, which is 0.5, and also the highest, 1000 microgram per ml. As you can see, there is no significant difference in the there's no significant difference between the treatment in Antarctica 3, meaning the cell in the presence of cycloheximide, the cell did not need adaptation period because it's slightly similar with the control. And for the it's very obvious for the tropical strain because at 1000 microgram per ml, they take about almost 10 days to start growing. So this one for this slide, I compare between the resistance strain, that highly resistant, which is the MIC can reach up to more than 1000 microgram per ml. That's why the temperate strain is not included here, because a temperate strain can only grow below 62.5. Okay. So next slide, please. Next slide. So for the general discussion, from the series of experiments conducted, it can be concluded that the strains originated from Sydney Island, and the one that didn't need adaptation period from the previous slide, we can consider that, and also strains from the Alaska, they are all showed very strong resistance towards cycloheximide. From the original habitat, which is soil of a harsh environment, might be the factor that caused Stichococcus strain from the Arctic and also the Antarctic to possess such an outstanding character. So from the Antarctic, we have 3 strains, and from the Arctic, 1 strain. So all of these strains were highly resistant to cycloheximide, while the tropical strain, which, if you still remember, they took a very long time to start growing in 1000 microgram per ml. So they're not as strong as the strains from the polar regions. So next slide, please. So one fact is that cycloheximide is actually originally was produced by a bacterium called Streptomyces gracius, in which
I am sorry.
Hello?
It's time. I am sorry. It's time to finish for you.
Okay, okay. I'm finishing. One fact is Streptomyces gracius, the one that originally produced the cycloheximide, can be found abundantly in soil, in which they produce cycloheximide and other antibiotic to reduce natural antibiotic. I mean, to reduce the influence of competitive. As we know, polar soil are very nutrient limited. Thus, the competition rate is high between the microbiomes. We can, we assume that the polar strains that can resist high cycloheximide, the presence of high cycloheximide until 1000 microgram per mil without any adaptation period, especially for the one from Antarctic, is because they might have been introduced to this antibiotic in their natural environment. So, and there are also reports that polar microalgae has been developed adaptive mechanism to thrive in the extreme environment. So what we can see from the cycloheximide resistance in Stichococcus might be the adaptive mechanism developed by Stichococcus, especially from polar region, to adapt the extreme environment. So thank you.
Thank you to you, dear colleagues. Have you any questions? Okay. I cannot can see any question at the chat. Thank you very much, Hiudayu, for your presentation. And we invite the next participant. It's Boris Donskoy. Welcome, please. Boris, where are you? Okay.
Можна виключити? 20 хвилин. Я зараз доповідаю.
Welcome.
Anybody hear me?
Yes.
Let's start. I'm sorry. Our idea was in investigation of immune phenotype of a member of Antarctic expedition.
Моя презентация.
But it's all okay?
No, we have you presentation?
Yes.
Yes, we unfortunately we cannot see it. Can you share it?
Yes.
Okay, thank you.
Oh, it's better.
No, yeah, maybe, yeah. But it should be the first slide. Yeah. Okay. Thank you.
Mm-hmm.
It's okay now.
Okay. Immune system, it's very complicated and very complex and extremely individual. Why it's so individual? Better question, for what? Immune system defends organism, but not know from what. The future enemy for immune system, it's always unknown. each day last million years, immune system fight with billions of bacterial, virus, fungi, hundreds of potential mutation, and immune system must save the body from this. But enemies are known, as I said, and immune system not have a general plan. Immune system must adapt for any enemies. It's impossible to be ready for anything. It's why if we ready for anything special, we lost another part of enemy. The theory about genetic variants and better or individual or more wild response good established in works of Ashley Moffat. Main idea is if you ready for something special, you always lost something other. It's why immune system of all our population is very individual. We have a better variant for response for something special, for example, for bacterial and virus, and not so effective response against another pathogen. We all have our Achilles heel, but our Achilles heel is another variant will be our strong side. It's very good established in individual HLA and cure combination, and resume it's optimum to survive infection, not good for reproduction. We are 25 years investigate the immune accentuation significance for reproductive outcome. And immune phenotypes must be absolutely not prognosed for pathogen. We all are very different. In this picture, you can see the photo of Olympic champions, and their body phenotypes make them champions. But the same body phenotype absolutely unfavorable for other type of sport. It means that we not have a normal immune system. All immune system have a more effective side and not so effective parts. What phenotype are favorable? Favorable, it's the phenotype that the same as your type of sport. It's more good picture because women have another variant of immune function. It's reproduction. What is for body is for? It's for running, jumping, cycling. We don't know what we are what we are meet in the future. One body type for all type of sports, it's impossible. What are most favorable phenotype for women? It's phenotype for reproduction, male attraction, childbirth, and we can't forget about bipedalism. You need to consider everything, every factors, and you can't always get what you want. Any immune phenotypes is more favorable for one condition and not favorable for another. Very good example is, for example, how we investigate the immune accentuation. The expression of CD8 on NK cells, nobody knows what does it mean and what a role of CD8. But we can see that population separately on 3 different phenotypes with low expression of CD8, high expression of CD8, and moderate. And we can see that in a population, the expression of CD8 is trend to 50-50. But what expression are more favorable? Favorable for something. We investigate for reproduction. we can see the expression of CD8 in patients in a day of embryo transfer. In this day, all these women obtain good quality embryo. We not see any difference in distribution of these parameters, but in a month, some of them became pregnant and others not became pregnant. This is a distribution not similar, and we can see that high expression more likely to patients that not became pregnant. And in 8 months, we can analyze that patients that have normal pregnancy outcome and live births generally have a very normal distribution of CD8. In the same time, patients with pregnancy failure often have a low level of expression. And elevated expression of CD8 significance for implantation failure, in the same time decreased level of CD8 significant predictive for pregnancy failure. We not obtain information about function of these markers, but we find the clinical significance of these markers. All type of expression, it's normal for health. It's not a problem, but for reproduction, it's unfavorable factors. It's unfavorable, but we can see some levels of CD8 also became pregnant and have pregnancy success. In the same time, a lot of patients with normal expression have a problem with reproduction because a lot of different another factors can work for reproductive outcome. But our idea is that normal, balanced CD8 expression is always good for clinical outcome, because you can regulate it, you can change it, you can increase it or decrease it if you need. In the same time, these patients can increase expression of CD8. It's like a favorable passage. It's not only possible way, it's a more easy way for better outcome. About Antarctic expedition, the same idea. With the same idea, we investigate all members of Antarctic expedition. Why our interest for these topics? Because it's a good models for investigation of immune phenotype significance for future state of health during expedition. We try to find immune predictive factor for unfavorable state of health, the impact of expedition on immune system, and try to find some changes in immune phenotypes in unusual environmental. We investigate 48 applicants for expedition. We study the immune subpopulation, lymphocytes, subtype of lymphocytes, granulocytes, the markers of activation of lymphocytes, KIR expression on NK and T in NT cells. We also investigate the NK toxicity as a basal innate immune defense, and serum level of IgG, IgM, IgA. And we froze all serum samples for subsequent comparison with the samples that we obtain after expedition. We also interested in investigation of level anti-infection antibody that can be changed after expedition. But we not expect about COVID. We investigate the members of expedition, future members of expedition, from December to February, and on February we obtained 70 positive samples. It means that near the half of members infected before testing, and one was infected after testing. Majority of individuals relapse in mild form or asymptomatic form. But anyway, we find some change in immune phenotype after infection. Absolute count of B lymphocyte was increased, and frequency of B B lymphocyte, and the level of activated markers isolate the air, and the level of regulatory NK cells was increased. It means that we have two groups clinical, and we must separate the members of who obtain COVID and who not obtain. Our previous data, we investigate absolute level of leukocytes, lymphocytes, and granulocytes. And we find the extremely high expression, extremely high level of lymphocytes and leukocytes in one individual. It's very bright accentuation, but this individual not pass the medical commission before expedition. It's good for expedition because we can wait a lot of surprise from this individual, but he not pass the medical commission. But we see not so bright, but also interesting accentuation in level of lymphocyte and granulocyte. The same, we investigate T cell level and T helpers level, and the same situation. In some individual, we have a rather deviated decrease of T helper cell. And we wait for clinical information about further state of health. Same situation we can see in expression of chronic inflammation markers on T cells and NK cells. In some patients, we can see rather increased expression of inflammation markers. And we also interesting, is it significant for state of health during the expedition or not? The same, we investigate the level of CD8 and NKT level. And also we can find some deviated individuals. Interesting that the same individuals often have the connected accentuation, increased level of NKT cells and increased level of NK cells express CD8. The absolute level of NK cells also down-regulated in some individual, and it's not normal for men. And we wait for information because it's very interesting. The same increase absolute number of NK cell, it's maybe good for health, but it's maybe also a response for inflammation. The same interesting distribution we find in NK cell toxicity. For example, this level, it's too low for healthy men, but in another situation, it's maybe normal for men after COVID-2. It's why it's a very big problem for us to analyze these groups as a whole, because people who was infected have some change in immune phenotypes. We also investigate the level of antiphospholipid antibody, and in one person we find a highly elevated level of antiphospholipid antibody. But this woman not member of expedition, and she also have positive for COVID antibody, and she has some fertile complication. But in two members, we find the moderate level of antiphospholipid antibody. Naturally, it's not means that it's antiphospholipid syndrome. It's only one incidence of antiphospholipid antibody positivity, but it's make a pro-autoimmune condition of this immune system, and we are also waiting for clinical information. Conclusion: we have groups where we detailly analyze the immune phenotype. Of course, we wait for it. Majority of individuals have a normal, healthy immune phenotypes without any accentuation. and we not wait a surprise from them. But some individuals have isolated or even systemic immune accentuation, and it's form a potential unfavorable state of health. Significance of accentuated phenotype will be investigated when we compare association of immune factors with clinical information during the expedition. We also wait for serum samples for comparison. Now we just on the beginning. However, the trip promised to be exciting because we hope that we obtain absolutely unique information about immune change during the expedition and significance of some immune factors for state of health. Thank you for attention.
Thank you to you, Boris. I saw the first question for you. Can you see the chat? If not, I will read for you.
Yes.
Such question?
No, I mean,
Answer, please.
Of course, immune system have a lot of homemade reaction and homemade mechanism, but that we obtain in evolution of million years evolution. It's a toll-like receptor, a lot of not specific mechanism, but I mean that immune system never know how strong must be reaction. And it's our problem. It's a problem of immune reaction because if it's not, if it's foreigner and antigen, immune system must respond. But its reaction must be extremely powerful or not so powerful. And it's a problem of immune reaction, the same as COVID-2. You must generate the immune reaction that enough for this battle, not more, because in another situation, immune reaction kill you.
Thank you very much for your answer. Have we any questions? Okay, please. You can speak. Okay, Masha, welcome.
Yeah, thank you. Thank you for the nice and very interesting presentation. I was just wondering, because you're a bit limited in time and you didn't have the chance to show the details of your research, but maybe I just missed it. Will you split your data in some further groups for some subsequent analysis based on some individual specific traits of these people? Because we know that some lifestyle traits, also the history of previous diseases and infections, and the history of some chronic diseases in the family could influence, well, potentially such sensitive data. Will you split these people on some groups and cohorts for some subsequent analysis?
Thank you for question. Its main problem of all immunological investigation when we investigate somebody with active immune inflammation. Immune system not looks like healthy if you have infection, but these groups are interesting because it's healthy individuals. At least they are healthy now. They are thinking that they are absolutely healthy and they are pass a medical commission. We must investigate the predictive or risk factor in time when it's not start to be realized. Because if the children have temperature, he have absolutely all immune accentuation, but it's not predictive, it's response. One day before, these children have absolutely normal, healthy immune factors. And we try to find if we can find something that are not start to work, but it was predictive for unfavorable clinical outcome. The same as we work with a healthy woman that have only one problem. They have subfertility. And because you are right. If we investigate the population, somebody have chronic disease, somebody have another variant, and the immune phenotype in these groups will be absolutely not comparable. And we must to observe the hundreds, thousands individuals to find something. Thank you.
Okay, yeah, thank you. Thank you.
Hey, dear colleagues, any questions? Okay, if no, I would like to thank you very much to Boris for such unusual presentation for Antarctic Conference, and hope for fruitful collaboration in future. Thank you very much once more. See you, bye.
Thank you.
Okay, and we will go to the next participants. It's Yaroslav Pirko. Is here or not?
Yes, yes.
Yes, okay. Nice you meet you here. Please make your presentation.
Okay.
Thank you.
Dear colleagues, let me introduce my presentation, Assessment of Colbanthus quitensis Genetic Diversity in Antarctica by Analysis of Intron Polymorphism of Cytoskeleton Protein Genes. Colbanthus quitensis is one of the two angiosperm species in Antarctica and has a very long longitude range, which in this region is represented by isolated island populations in a wide range of conditions, which might the result in the appearance of many and various ecotypes in the region. Insufficient attention has been paid to the study of genetic differences between Colbanthus quitensis populations. But information on its genetic variability remains so far quite sparse and based on mostly on results of only a few studies encompassing a geographically limited set of sample populations. Thus, a study of genetic differences between the populations of Colbanthus quitensis in different environmental conditions employed the contemporary palette of molecular genetic markers becomes an urgent task. We studied with exons of highly conserved housekeeping genes, for example, cytoskeletal proteins genes, such as actin, alpha, beta, and gamma tubulins, are represented in different organisms and genomes by homologous sequences, and introns belong to hypervariable regions of genes and can have different lengths. You can see in this picture the different lengths of introns by as example of intron exon structure of flex beta tubulin genes. The analysis requires a regular polymerase chain reaction, PCR, using primers to exons, which flank the introns. Thus, allowing one to use a single combination of degenerate primers for any plant DNA. A serious advantage of the method is the fact that the analytical system does not require previous information about specific gene sequences of the studied plant, as the primers can be developed for species with sequenced genomes with further duplication to any other species, since the exon sequences of the selected gene are highly conservative in all plants. Many variants of ILP markers have been developed based on many housekeeping genes, enzymes of the electron transport chain, and various key structure proteins. In recent years, markers of intron length polymorphism have been successfully used for genetic mapping, species identification, and large-scale genotyping analysis. ILP markers have been tested time and again on various objects. The possibility of applying them to molecular genetic analysis of plants has been examined not only for species but also for cultivars. One of the most successful ILP markers today is considered to be the method based of assessing the intron lens polymorphism of beta-tubulin genes, TBP method. The TBP method is an universal approach that allows genotyping and genetic differentiation of various plant species, varieties, and genotypes based on knowledge of the exon intron structure of plant beta-tubulin genes. The molecular markers of this type have been previously used to reveal and analyze the level of genetic polymorphism of the Antarctic hair grass in previously our publications. Since the TBP method has demonstrated high efficiency and in order to expand the palette of ILP markers, new DNA systems have been developed. New markers are based on the analysis of the intron lens polymorphism of other cytoskeleton genes such as actin, alpha, and gamma tubulin genes was done. In this picture, you can see these markers. The aim of the study was to identify molecular genetic differences between different populations of Calabanthus quitensis in Antarctica by estimating the intron lens polymorphism of alpha, beta, and gamma tubulin genes, as well as actin. Using the Calabanthus quitensis from different natural populations of the Argentine island region collected during the 24th and early Ukrainian Antarctic expeditions. You can see the regions of our investigation in this picture. In this picture, you can see two electrophoregrams for alpha TBP and gamma TBP. Electrophoregram, some words about this electrophoregram. Analysis of the intron lens polymorphism of genes coding alpha and gamma tubulin produced quite a few amplicons. You can see a few amplicons of alpha tubulin genes. We also found that at this point, data on ILP in genes coding alpha and gamma tubulin do not allow efficient genotyping of samples of Calabanthus quitensis and consequent differentiation of island population. But intron lens polymorphism analysis of gene coding actin revealed low level of genetic polymorphism in Calabanthus quitensis by this marker in the studied region. In particular, there was found intron polymorphism of the actin genes at the intrapopulation level among the specimens from the core island populations. In this picture, you can see B. The molecular genetic differences between natural Calabanthus quitensis populations from the coast Graham's land were also revealed by assessing the second intron polymorphism of beta-tubulin genes. In this picture, you can see in this field. Among all studied populations, the population from island Booth, in this picture H, which may be a consequence of a changes in the existence conditions of these species. Okay. We also obtained the electrophoretic spectra of amplified fragments of the beta-tubulin gene, the first intron, and we have some results which demonstrated the low level of genetic polymorphism, Quitensis, Colobanthus quitensis, in Antarctic was shown by the intron length variability of the cytoskeleton protein genes. Thank you for your attention.
Thank you, Yaroslav. Probably some question to Yaroslav after this presentation. I need to check the chat. Okay. No question to Yaroslav. This very interesting presentation about Colobanthus quitensis. And we need to go to another points of. We can continue, Olena?
Yes, we can continue.
Okay, thank you. So, Iryna, probably we will need to start the poster session because no paramono. Okay, I have information that first 10 presentation we have now, and we can start with Dr. Abu Bakr.
Hello, yes, I'm here. Can you hear me?
I only inform that we have 15 minutes for such presentation. So if you want a question, 10 minutes for presentation and 5 for the question, okay?
Okay.
Go ahead.
Okay, so I'll share my screen now. So can everyone see my slides?
Yes.
Okay. So my name is Nurliza Abu Bakar, and I'm a PhD student from Universiti Malaya, Malaysia. So today I would like to present part of my PhD work titled Heat Stress Response of Pseudogymnoascus species isolated from different biogeographic regions. Before that, I would like to thank my collaborators, which are not here today, Dr. Benjamin from the MPOB and Dr. Jurek from the Polish Academy of Sciences. So as a start, I would like I would like to start with general information on the soil fungi, as how my colleagues Dr. Adila and Dr. Rizman has explained yesterday. Soil fungi can be used as a sensitive indicator for soil health and quality. So with the knowledge of the mechanism of heat stress response, we hope that it can contribute to the understanding of the specific roles of soil fungi in the extreme environments. In this work, we use 6 Pseudogymnoascus species that were isolated from 3 different regions, 2 from the Arctic, 2 from the Antarctic, and another 2 is from the temperate region. So why we are choosing Pseudogymnoascus? Like I said yesterday, Dr. Adila and Dr. Rizman has presented deeply why, and mainly it's because they are commonly isolated from various regions, especially the both polar and temperate region. They have high similarities in the temperature-dependent growth profile between the species. So these we think that they are good candidate to search for heat stress biomarkers in soil fungi. Okay, the research questions that we would like to answer in this work is 1. Under heat stress, is there any similar heat shock proteins that were upregulated in all isolates?
And probably the second one, if we can find any potential biomarker for heat stress in Pseudogymnascus species. However, for this poster presentation, my objective, I will put it as in one objective here, which is to determine changes in the protein abundance within different strains of Pseudogymnascus from polar and temperate regions in response to heat stress. So the diagram here showed the simplified version of methodology used in this work. We use tandem mass spectrometry to profile protein extracts of Pseudogymnascus species, and then later with the results, we create we identify after we identify the protein, we create the proteomic profiles of each isolate and then compare them according to their region. Okay. I will show only part of the results that I got from this work because it involves 6 isolates. So figure 1 show the number of proteins that being up and down regulated in each isolate. So as you can see here that all isolates show different total number of proteins that were up-regulated and down-regulated. So they show a variation in the number of proteins involved. With the protein identification result from figure 1, we analyzed the protein using gene ontology enrichment analysis of only the significantly upregulated differentially expressed proteins, and figure 2 showed the top 10 pathways of all isolates. The top one is the Arctic isolate, the middle one is the Antarctic, and temperate. The bottom one is the temperate isolates. So I think you can't really see each pathways in this slide, but all isolates showed diversity in their response of upregulated protein in response to the heat stress. However, there are also common proteins and common pathways that were found in all isolates. Okay. So in this figure, it is a simplified diagram of the 3 major mechanisms used by Pseudogymnascus. So, like Dr. Adila presented before, she also found through transcriptomics that the protein homeostasis and DNA repair are part of the major mechanisms used by the fungi in response to heat stress. So the first mechanism used by Pseudogymnascus in this work involve protein homeostasis, such as the urea cycle, various amino acid metabolisms, the protein transport from Golgi to ER and vice versa, and also ubiquitin proteasome pathways. Okay, the second mechanism is the energy production that involves various carbon, glycosylate, dicarboxylate metabolisms, and also pentose phosphate pathway. All isolates shows an upregulation of protein in the TCA and electron transport chain. And the last or the third mechanism involved in the response, in the heat stress response, is DNA damage and repair, which were presented by a few pathways such as RNA degradation and nucleotide salvage pathways. However, there are also a few other pathways that are upregulated in all isolates, such as the general cellular response to heat stress using HSF1, and then cellular response to external stimuli. There are also a few of the isolates produce or upregulated biosynthesis of secondary metabolites and antibiotics, and also activated their innate immune system. Okay, so for a conclusion, the Pseudogymnascus use various molecular pathways in response to heat stress, And there are 3 major mechanisms contribute to the survival of 6 pseudogymnoscus isolated from various regions examined in this work. Our proteomic analysis identified no significant relationship between responses observed and biogeographic origins, regions of origin of the isolates. Okay, this is the what we can do for future work is to increase the number of isolates used in this work to be able to say that they represent each or one or the biogeographic regions. Okay, because in this work we can only use 2. So it is not really, we can't really see different or significant relationship between the responses and the biogeographic regions of origin of each isolate. Okay, because I was told that we have only 5 minutes presentation, and so that's why this is a very quick presentation. Before I end, I would like to say Selamat Hari Raya to all my Muslim and Malaysian friends, and happy holidays to everyone in Malaysia. So that's all from me. Thank you.
So thank you for your interesting presentation, Nurizin. I don't see question for you. Okay. And we can go to the next speaker, Rasidni. Rasidni.
Yes, I'm here.
I'm here. Oh, very nice. Go ahead.
Okay, thank you. I shall share my slide. Can everyone actually see my slide?
Yes.
Okay.
Full screen, please.
All right. So this is fine?
Yes.
Okay, thank you. So good morning or good evening, everyone. My name is Rasidni, and today I'll have a very brief presentation on a tiny part of my PhD study, which is the Antarctic Bacteria Consortium diesel degradation optimization via statistical approach. So the objective of this part of my study is to maximize bacterial diesel degrading capability by studying the effects of biotic and abiotic factors. So the factors that will be manipulated respectively are salinity, nitrogen source, nitrogen concentration, pH, temperature, and also diesel concentration. So in this study, the optimization will be carried out using bacterial consortium, noted as BS9, sampled from soil in Bernardo O'Higgins' space by the Chilean research team. So a very brief result of my study so far. So as for salinity, the bacterial consortium actually preferred 0% salinity. So this could be because the bacteria was sampled from soil, which is at an area nearing the diesel fuel tank instead of coastal area. And as for nitrogen source, the bacteria actually preferred ammonium chloride, as it's shown the highest degradation efficiency at almost 80%. And as for concentration, the bacteria works best at 1.0 gram per liter nitrogen concentration, as well as the growth, which also peak at 1 gram per liter nitrogen concentration. And next, as for pH, the bacteria degrades hydrocarbon best at a neutral pH, which is at 7.0. However, as you can see at the red curve, the growth of the bacteria keeps on increasing, and it actually prefers a more alkaline environment. But because the main objective of this study is to identify the best degradation efficiency. So pH 7.0 is noted as the best factors for it to degrade hydrocarbon. So next, as for temperature, the degradation efficiency was high at around 10 to 15 degrees Celsius. And lastly, for diesel concentration, the degradation efficiency peaked at 3% of diesel. But as you can see at the red curve as well, the growth of bacteria, it has high tolerance towards very high concentration of diesel. And even at 7% of diesel, the bacterial growth is relatively high at around 1.0 from spectral photometry analysis. So to conclude this very brief presentation, after optimization, almost 91% of diesel was degraded within 7 days of incubation. This is slightly higher than pre-optimization, where during the screening process, only around 80% of diesel was degraded. So this is this short summary of this result that I've shown just now, where the best parameter for bacterial consortium BS9 to degrade diesel is at 0% salinity, 1 gram per liter of ammonium chloride as nitrogen source, pH 7, and at 10 degrees Celsius as the best temperature. So these are the references that I've used in the making of these slides and also part of my research. Thank you so much.
Thank you so much too. Are some questions for our Sydney? No question. I need to check chat. Okay, I have one small question. So we can use this experience in some bioreactor in Antarctic condition. What do you think about this?
Yes, actually that is part of my study, but it will be carried out somewhere later because I'm still at early phase of my study actually. So I'm still at the optimization stage, but after this, we will try to apply it in bioreactor and to try to apply it in Antarctic as well.
Yeah, because practical aspect is very important. Thank you very much.
Thank you so much.
I invite next speaker. Unfortunately, I don't know name. Tenku Mazuki. Effect of heavy metals on phenol degradation by an Antarctic bacterial consortium. Oh, okay.
Okay. Hi.
Go ahead.
I'm here. Okay. Okay, I will share my slide. Okay. Hi. I'm Tengku Atira Mazuki, a postgraduate student from Universiti of Malaysia, and today I will present part of my study, which is effect of heavy metals on phenol degradation by Antarctic bacterial consortium. So, moving on to the introduction, as we can know, and as we know, Antarctic is not free from environmental pollutants, although it is often perceived to be the last pristine continent on Earth. In Antarctica, the presence of heavy metals by natural sources and anthropogenic activities needs to be considered for degrading phenol compounds. And exposure to heavy metals creates a stress condition for bacteria and disrupt their metabolic reactions. Therefore, this can be a major challenge for bioremediation of pollutants, including phenolic compounds. So, moving on to the hypothesis, which is the bacterial community obtained from Antarctic soil can degrade phenol in low temperature and will not be hampered in the presence of heavy metals, making it viable option for in situ bioremediation for phenol in Antarctica. So this is just a simple experiment, which is for metallurgy, the samples collected from Antarctic expedition will be preliminary screening for consortium for phenol regarding activities, and then the consortium will be introduced with 1 ppm of heavy metals in the optimized media. So moving on to the results. So this is the result of the effect of heavy metals on on phenol degradation. And the bacteria growth was inhibited by 1 ppm of silver compared to other metals, and because silvers exhibit antimicrobial activities against a broad range of microorganisms, which will react with nucleophilic amino acids residues in protein and attach to phosphate and carboxyl group of membrane of the enzyme proteins that leading to protein denatured. So amongst of the 7 heavy metals, all the heavy metals shows the 100% of degradation of phenols except for cobalt, which only show the degradation was 78%. This is still maybe the cobalt disturb the cause of redox reaction that disturb the enzymatic reaction, and it will be suppress the ability of the bacteria to degrade phenol, and this can slower the rate of degradation. So moving on to this is just a brief experiment. So I move on to the conclusion. So the optimization of the media required to achieve maximum phenol degrading activities was attempted through the 1 factor at a time, and degradation was confirmed in the presence of 1 ppm of 7 different heavy metals, which the bacterial growth was inhibited by this ion in the rank order of aluminum, arsenic, cobalt, lead, nickel, chromium, and lastly is the silver. The greatest bacterial growth occurred in the 1 ppm of aluminum, achieving an optical density of 0.985, and lowest is 1 ppm of silver, which with an OD 600 of 0.097. However, while Ag has a considerable inhibitor effects of the bacterial consortium's ability to degrade phenol, 100% removal of phenol was achieved in the presence of the other metals ion. So this pilot study highlights the different influence of the different metal ions on the migration of phenol by native Antarctic bacteria. So that's all from me. Thank you.
Thank you too, Atira. Are some questions for this presenter? In chat, no. Maria, go ahead.
Yeah, thank you for your nice presentation. It was really interesting to listen. I just wonder, because I don't really have experience of working with metal resistance in microorganisms. Therefore, I wonder what was the rationale behind the choice of this dose of toxic metals, this 1 ppm. Did you test different doses, or is it from literature?
This is from based on other paper that shows 1 ppm of. I chose this one range of 1 ppm of heavy metals based on the literature review I read that from heavy metals literature review on in Antarctic. So that's so after this I will try to test a different concentration of different heavy metals to see whether it can go up until it can go reach more than 1 ppm or it's inhibited by the 1 ppm. Okay.
Okay, yeah, thank you very much.
Thank you.
Yes, I want to ask a similar question. Biodegradation of phenol, yes?
Yes.
In conditions of presence of heavy metals, yeah? So in reality, what pollutant you mean? For what pollutant you prepare this this scheme of biodegradation?
I'm sorry. Do you mean in reality in Antarctic?
Yes.
Or, okay.
do you plan to do you plan use this technology for some concrete pollutant in Antarctica?
Yes, I try to use this phenol as in Antarctic there is pharmaceutical sewage that is being discharged into the discharged from the station. So I will try to use this to, as you know, pharmaceuticals discharge usually contain phenolic compounds. So that's why I choose this phenol pollutants to see whether these bacteria can be used in sewage, in pharmaceutical sewage in Antarctic to degrade phenolic compound.
Okay, thank you. Thank you for presentation. The next speaker was probably title Growth Evaluation of Antarctic filamentous algae in molybdenum as heavy metal.
Hello, can you hear my voice?
Yes, yes.
Thank you. Can everyone see my presentation?
Yes.
A very good morning and good evening to everyone. My name is Valeria. I'm from Universiti Putra, Malaysia. The topic of my poster presentation is Growth Evaluation of Antarctic filamentous algae in molybdenum as heavy metal. So heavy metals, there's a lot of heavy metals have been reported in Antarctic region. And this is mainly because of the anthropogenic sources, such as weathering process from the oil spill incidents, waste disposals, and the heavy metals also can be transported through biological systems, such as through penguin or seals, as well as the long-range transportation of global contaminants through the Southern Ocean. So these heavy metals can cause various toxicological effects. So in my study, I choose molybdenum, which is a trace element which toxicity to the Antarctic ecosystem has not been thoroughly investigated. And this heavy metal can be persist longer in the colder environment when exists in a large quantity. So as we know that the, for information that the Chile is the one of the world's second largest producer of molybdenum, which is close proximity to the Antarctic region. This may pose potential risk of molybdenum pollution to the Antarctic ecosystem. And molybdenum is commonly found in the oil and automotive industries. And for the past 10 years, there have been large quantities of molybdenum mining has been done for the economic production. So for molybdenum reductions by using Antarctic bacteria have been previously reported and shown the ability to reduce into less toxic form. However, most abundantly available algae in the Antarctic have not been previously utilized for bioremediating molybdenum. Therefore, in my study, I'll be focusing on using freshwater filamentous algae to bioremediate the molybdenum. So for the methodology, I'll be cultivating the Capsomodius species, which was previously identified in the Odesa media with two different aeration systems, with and without aeration system. These samples were isolated from the Greenwich Island, which is at the Antarctic Peninsula, followed by the optimization of the various parameters such as carbon, nitrogen, and carbon concentration, salinity, and pH, which was cultivated with 10% of algae medium in a low phosphate medium, which was subjected with 10 millimolar of sodium molybdenum. This was then the optimized parameters will then be further analyzed in a simple exponential growth model. As for the result, from the standard growth curve, it was known that the day 3 was the exponential phase for the algae, and in the specific growth rate of the Capsomodius species was much higher in the aeration system compared to without aeration. In the next OPET, one factor at time analysis, the highest biomass was produced from the sucrose of 20 gram per liter, ammonium amount of 2 gram per liter and salinity with 4 gram per liter at a pH of 7.5. All of these optimized conditions will then be further implemented for the growth kinetic analysis. So based on the growth exponential model, it was known that the both growth rate in the aeration and without aeration system was not substantially different as per the exponential growth model. So therefore, to conclude my presentation, this study will act as a preliminary study for a molybdenum reduction by algae, and also different models, growth models can be used to compare with the existing exponential growth model. That's all for me, and thank you.
Thank you too, Hayat-Hi, for very speech presentation. Yeah, I'm really very excited that the ACLIMA have so much different branches of biodegradation branches. So it is probably a little bit difficult to prepare question, but I probably somebody have question. For me, it is very interesting why algae, why green filamentous algae in comparison with bacteria. Why will you use algae for such approach?
First thing, because one is mostly abundantly available, and previously there have been report have been using bacteria. So we try to use algae to bioremediate the molybdenum, and also in different study, algae also has the ability to degrade oil contaminants. So might as well the algae can has the potential to reduce the molybdenum.
Okay, thank you for your answers. Thank you for your presentation. Good luck in this very important thing.
And we will go to the next person. It's Victoria Ivanets. Victoria, are you here? Victoria?
Please unmute your. Okay.
I'm here.
Yes, yes, this one. Okay, Victoria?
Yes, is the time visible?
Okay, yes, it's time of your presentation.
Okay.
Join us and welcome.
Is it visible?
Yes, everything okay.
I could start, yes?
Yes, we could start.
Hello to everybody. I want to introduce my presentation. Let's name an example of ornithochory of Cataracta mccormicki in the region of the Argentine island of the maritime Antarctic. In this work, we examine the specific using of Cataracta mccormicki. This another name is South Polar Squaw, using plant materials, including plants in the Argentine island region, as example as a possible ornithochory in terrestrial ecosystem of Antarctica. I want to mention this ornithochory. It is a distribution plant of seeds of plants by birds. It is special case of the zoochory. Skua is a bird which widespread throughout coastal region of Antarctica. They nest all around continental Antarctica and breed into the deep south. The samples when we collected and worked it is Argentine island region. It is on the map, and they included itself Argentine islands and also islands that are nearest. It is from north to south. It is from Bus to Barcelona, and from west to east from Cruz to coastal area. Also in this area it is located Galindes Island, where is Ukrainian research base is located, Academic Vernadiki. The total number of analyzed nest of Cataracta mccormicki was 186, and they were collected during Antarctic summers during 2009-2020 years. On this slide, it's showing the main types of the Cataracta McCormickii nest into region of Argentine Islands. For our investigation, we divided component of nest of this bird by through main components and calculated the percentage of every component in the nest in all season. We divided on such groups like mosses, lichens, limpets, feathers, bones, terrestrial green alga Praseola crispa, and high grass Deschamps antarctica. On the photo A, it is showing the main popular nest of Cataracta McCormickii in the region. It is based on the moss bank. It is also included a lot of mosses inside, which is in the region. It was like maximum 82.2%. On second photo number B, it is showing nest with lichen component. Lichen components in the area was not too much, up to 20.5%, and in this nest also it is lichens named Umbilicaria, also quite rare in the region. On the photo D, it is nest of Cataracta McCormickii with Deschamps antarctica inside and around, and it's showing big population around where this nest is located. Also in the nest was found Praseola crispa. It is green algae. In this amount was up to 5.3% at maximum. In some pool with some island with poor vegetation like Grotto was up to 60% into nest. And also some minor amount of feather and bones was also found, maximum 1%. And also very interesting example of nests were found on the island named Locator in the region when South Poloscura was nesting on the bare rocks without using any vegetation, any plant materials. On this next slide, I want to show the diversity of mosses that was found in the nest. And in our investigation for 4th season, we analyzed contents of moss fraction and found that 4 mosses was included in all season in the nest. It was Polytrichum strictum, Saniona species, Pohlia nutans, and Brium species. Also, these 4 mosses are typical to the most popular moss banks of the region of Argentine island, which named tall moss turf subformation, whereas Polytrichum strictum is dominant, and showing on the photo up. And Bryophyte, mat, and carpet formation showing in down photo. It is the Saniona species, the main component. Birds nesting mostly on this territory into area because it is the most comfortable, the most hot places. They opening during the breeding period of the schools that started from the end of November and first 10 days of February. Mostly in the, as I said before, into our region, mostly they make a nest on such on moss banks. And also on the down photo here, it is showing the Deschamps antarctica, which in the region are included also Bryophyte, mat, and carpet formation. And we studied separately if there are this, hair grass included into nest or not. And nest with Deschamps antarctica through all nest was 9%. And very important to say that mostly all nests that included Deschamps antarctica inside was a lot of Dechampsia around the nest, and it was mostly bigger, very big old population around. That showing us that birds probably choose something for the nest that they located not very far. And the same possible to say about mosses, as I forget to say, that also through analyzed by species of the mosses was included into nest, we also understand that it is the most typical of moss bank component, and that's why we make suggestion that conclusion that birds choose the most common materials from around the nest. And the conclusion, I want to say that in our study we didn't observe something that indicates that South Polar Skua choose certain massively prevailing materials to build their nest. What is a very good correlate with the literature data from the more north regions. Also, we suggest that in Argentine island regions, South Polar Skua are not selective while choosing the nesting materials, and also we didn't find support. No, we didn't observe that South Polar Skua spread higher grass deschamps Antarctica between different islands or coasts, but we suggest that it could distribute some vegetation components locally, thus furthering the ornithochory of plant. That's all. Thank you for your attention.
Okay, Victoria, thank you very much for your presentation, especially for so nice photos, because the most presentation we could see rocks and ice, and from your presentation, such a special photo which so like our eyes. Thank you very much. Okay, dear colleagues, have you any questions? Let's, it seems to me chat too. Okay, no. Okay, thank you very much to you, and we will go to the next presentation. Our colleagues from Belarus, Gigignak, our colleagues, have we?
Вроде присутствую, слышно?
Так, да, конечно, мы вас... one moment. I should explain, it's our colleague from Belarus, and we have such special agreement. Dr. Gigignak will present his presentation in Russia. Excuse us, if you will have your questions, please write them in chat, and I will translate from Dr. Gigignak, and I will translate his answer. Thank you for understanding the situation, and I invite Dr. Gigignak to present his result. Пожалуйста, да, мы просим вас представить ваши результаты. Спасибо.
Добрый день, спасибо за то, что разрешили участвовать в конференции и разрешили выступить не на английском языке. Но для девушек из Малайзии я отдельно потом переведу свой текст, и они, наверное, это поймут. И хочу выразить удивление прекрасному полу украинцам и девушкам из Малайзии за то, что они представляют большую половину всех докладов, которые участвуют на этой конференции. Это очень похвально. Но вашему вниманию я предлагаю небольшое сообщение о определении температурной выживаемости. Я сейчас попробую включить презентацию. Но у меня не получилось. А вот она получилась, да?
Ми поки не бачимо, ми поки не видим, но я готова, если что, и вашу презентацию расшарить.
О, это хорошо, если вы поможете мне сами представлять, пожалуйста.
Хорошо, давайте, я сейчас расшарю.
Да. Я пока буду говорить. Температура для, в антарктических озерах...
Видите презентацию?
Да, да, спасибо, да, все отлично, да, спасибо. Температура... Для определения температурной выживаемости одного из эндемиков антарктических озер, Ктенодафния студера, раньше она называлась Дафния студера, она обитает в данном районе, где мы исследовали, это Ларсиман Хиллс, Восточная Антарктида. Из 150 озер, которые там находятся на этом полуострове, вот вы на слайде видите, где это находится, она обитает в данном районе в 10 озерах. Эти озера по своей систематике, по своей гидробиологии очень различны, так и даже по возрасту. Так одно из озер, которое находится в центре этого полуострова, озеро Ареет, ему более 130 тысяч лет. И вот по одной из версий именно это озеро может являться центром, откуда может эта дафния распространиться по другим озерам и вообще в Антарктике. Но это одна из таких теорий. Температура для паикилотерных организмов является важнейшим абиотическим фактором, способным лимитировать их жизнь при всех других, даже оптимальных условиях, наличие корма, отсутствие хищников и благоприятных условиях. Но вот эта дафния, которую мы выбрали в озерах, она является самым крупным объектом животного мира, который обитает в этих озерах. Поэтому этот гигант этих озер и пристальное внимание сейчас подвергается изучению. В природных условиях гидробионты могут подвергаться быстрым и порой непредсказуемым изменениям температуры. Для широко распространенных видов клядоцера, а именно эта дафния относится к клядоцерам, к ветвистоусым ракообразным, практически ни у кого не определены значения верхней температурной границы толерантности. Для чего это нам надо? Ну, во-первых, мы все сейчас говорим о глобальных изменениях климата, даже в сторону потепления. Но, слава богу, Восточная Антарктида, наверное, менее подвержена этому влиянию. В этой связи нами были проведены эксперименты по определению температурной выживаемости этого вида. В пресноводных озерах, в местах наших исследований, это в районе российской станции «Прогресс», температура воды, вообще-то в литорале, если когда меряешь температуру воды в литорале, то она достигает порой даже до 13 градусов с плюсом. Хотя обычная температура меньше 4 градусов. Но если озера, которые пресноводные озера в данном районе есть глубиной до 30-35 метров, то, естественно, там на глубине температура будет держаться все время постоянно и не будет достигать таких величин. Сейчас так. Второй слайд, если можно, да. Здесь на вашем экране видите представителей вот это именно ктенодафния студери, эндемика Антарктики, на различных стадиях ее развития. Она выдерживает эти температуры, которые в течение, наверное, уже тысячелетий, и практически максимальная температура, которую она чувствовала, так сказать, на себе, это вот 13 градусов. Это в литорале. Вот. Но тем не менее, вот мы, когда начали... Следующий слайд, пожалуйста. Это дафния сама в озерах. Когда подходим к озеру и простыми гидробиологическими способами, если брать пробы, то мы увидим, что в одном кубометре воды может быть 1-2 таких дафний. Когда мы начали исследовать это с помощью подводных аппаратов, типа гнома, типа экшн-камеры, так как прозрачность очень хорошая и большая, то увидели, что все эти дафни, миллионы, миллиарды, все сидят на дне и на своем пищевом объекте, это водорослево-бактериальные маты там, а в толще воды практически их нет. И при определении температуры порога существования этих антарктических дафний и некоторых морских гидробионтов, таких мы использовали также в экспериментах морских амфипод, которые обитают при -2 градусах в данном случае, мы использовали самодельный переносной, можно следующий слайд, переносной прибор. Нет, мы пропустили, наверное, один с прибором. Там, посмотрите, пожалуйста. Вот этот прибор, да, пожалуйста. Вот это самодельный прибор, который основан на использовании элемента Пельтье. Элемент Пельтье – это термоэлектрический преобразователь, принцип действия которого основан на возникновение разности температур при протекании электрического тока, позволяющий получать как высокие температуры, так и низкие, вплоть до замерзания воды. Использование этого элемента нам позволяет не использовать разные антифризы, а только источник питания, электрический ток, и мы можем получать температуры вплоть до кипения, но мы до такого не доводили, и вплоть до замерзания. Такая, пожалуйста, теперь график еще один температуры, вот это, да, покажите, да. Здесь вот мы видим, в течение 3 месяцев мы измеряли температуру воздуха и температуру воды. Вот и синяя температура внизу, такая диаграмма, это температура воздуха, а все эти точки разноцветные, которые помещены на графике, они обозначают в данный момент температуру уже самой воды. И вот достигала температура в основном больше, около 10 градусов, это в летний период, а редкие только при ясной солнечной погоде достигала уже до 10-13 градусов. Вот, теперь, пожалуйста, следующий слайд. Еще. Вот здесь, да. На дальнем слайде приведены результаты экспериментов по определению температурных границ существования вот этого эндемика. Отметим по определению нижнюю. Здесь мы проверяли, в экспериментах мы измеряли скорость подъема температуры с разной скоростью, от 0,3 до 2,5 почти градусов в минуту. Одни эксперименты у нас были 8 минут, 10, 75 до 100 минут. И отдельный эксперимент, у нас вот третий слайд по горизонтали, мы охлаждали до какого-то периода, потом опять поднимали до температуры, при которой уже была для них летальным. Следует отметить, что вот эти... Если для них была летальная температура около 33, 35, 34 градусов, то даже при практически полном мерзании в полукристаллические такая уже вода находилась, отрицательная температура, они все-таки оставались живы. Это удивительный такой объект. Но они, наверное, такого никогда и не испытывали на себе. Но вот эти высокие температуры, нам было интересно узнать, при каких температурах высоких они могли жить там день, 2, 10, там, 20 дней. И вот оказалось, что при температурах около 25 градусов эти дафнии спокойно живут и размножаются при 25 градусах. И, пожалуйста, тогда следующий слайд. И вот нами впервые, до этого мы определяли максимальную температуру выживаемости для десятков гидробионтов, как обитающих в озерах там, Белоруссии, в России, везде, и определяли температуру для креветок, для камчатских животных, которые обитают в геотермальных источниках. И вот с применением вот этих всех данных, это вот такая усредненная данная, мы получили уравнение зависимости верхней летальной температуры от температуры среды обитания, вот на примере таких низших гидробионтов. В температурных пределах их существования практически от минус 7,5 градусов до 39-41 градуса. Ну, я поясню, что, например, минус 7,5 градусов это дюналиэла, водоросли, которые обитают в соленом озере Балпаш при температуре 7,5 градусов. Там соленость до 350 промилле. Вторая точка – это гомарус из Антарктики, как раз из моря Содружества, при минус 2 обитает. Затем идет наша дафния. Я брал только такие знаменательные, так сказать, точки, которые эффективно показывают, достоверно все показывает. 4-я макробрахиум непонензе. Это точка, которая для субтропической креветки макробрахиум непонензе, обитателя Восточной Азии. И 4-я точка, уже потом у нас будет идти, лимнея ходутка, которая нами определена впервые. Это новый вид для науки, обнаружен нами на геотермальных источниках Камчатки, которая выдерживает температуру до 41 градуса. Как видно, из полученной нами зависимости существует какая-то общая закономерность для гидробионтов, обитающих практически во всем температурном диапазоне их существования. Вот. Ну, я не буду больше так говорить, но будем надеяться, что наши озерные обитатели, антарктические, частные местные эндемики, такие как вот эта птинодафния тудари, выживут при любых глобальных изменениях климата. Вот. Спасибо за внимание, но я хочу сразу отметить, что мы надеемся, что и вы примете участие в нашей конференции в Беларуси, которая будет у нас в сентябре где-то, в середине сентября мы пришлем вам обязательно приглашение. И ну, будем проводить, конечно, так, как и вы проводили, онлайн. Спасибо за внимание. Thank you for invitation.
Okay, thank you very much to you for your presentation. Можете сразу сказать дедлайны вашей конференции? Где можно посмотреть информацию? Вы ссылку можете сделать буквально в чате. Dear colleagues, Dr. Giginyuk invited all participants of our conference to take part in Belarusian Antarctic Conference, which will take place in September? In September? Okay, and I asked him to provide us more information about this event, and we will share this information through all participants of our conference. Вы поняли, что я, да, все это перевела и сказала, что вы приглашаете, и что мы эту информацию расшарим, ну, в общем, разошлем всем участникам нашей конференции. Хорошо?
Да, да, конечно.
Okay. Thank you very much once more. Have you any questions, dear colleagues?
У меня есть.
Okay,
Юрий Григорьевич, я рад вас.
Слышу у тебя, что?
Юрий Григорьевич, рад вас приветствовать. Да, рад приветствую вас, рад видеть. У меня к вам вопрос. Меня интересует вот глубина озера. Так, вот на какой глубине вы находите дафни и наблюдается ли вот какой-то градиент температур от поверхности до максимальной глубины?
Да, самая максимальная температура в этом озере, это озеро Прогресс, там температура 35 метров. Вот. Остальные озера, они более неглубокие, там от 4-5 метров и до 35. А практически все летний период, они, кроме одного озера, Болдер, который вечно подо льдом, они открываются на месяца 3. И здесь градиент температуры только уже на 2 метрах температура плюс 4. Вся литераль, одна только литераль прогревается. Поэтому здесь нельзя говорить, что дафни все время, гидробионты, которые обитают там, чувствуют эту температуру на себе. Уже на глубине они этого не воспринимают. Поэтому говорить, что какой-то градиент температуры, да, мы мерили. У нас такой прибор есть, Хариба, как вы знаете, может, знаете. Мы его гидрохимию и всю температуру делали по вертикали. И термоклина не обнаружили, но уже буквально 1-2 метра сверху уже идет плюс 4, плюс 3 градуса. Такая температура.
Понятно.
Ну, а по то, что они обитают на дне, это я вот уже сколько, 3 раза уже был на этих озерах, работал, и никак не мог понять. Я беру обычными способами эту гидробиологию там, по сетками и батометрами, и не мог найти этих дафни в хорошем количестве. И вот когда мы запустили подводный аппарат «Гном», и когда мы уже экшн-камеры начали опускать, то мы увидели, что все эти козявочки спокойно себе миллионами сидят и кушают этот водорослево-бактериальный мат, который, в общем-то, в изобилии, хотя и не такой толщины, как хотелось бы, да, этот водорослевый мат, свет проникает сплошняком до 35 метров, и он активно зеленый, вот вы видели такой горбик, такой был на слайдах, и толщина его всего лишь ну, полсантиметра, не больше. В основном ниже уже идет уже, видимо, вот вы справа такие горбы этого мата. И вот они сидят и кушают здесь. Развиваются они активно, причем даже когда мы испытывали их при высоких температурах, вот они сидели у меня 25 градусов около 2 месяцев, они спокойно размножаются, дают эфипиумы, то есть можно наблюдать их весь жизненный цикл. К сожалению, жизнь в этих озерах ограничена видовым разнообразием. Так как я сказал, это самый крупный живой объект, который находится в этих озерах антарктических. Ну, еще, наверное, в Западной Антарктиде там есть личинка комара, но в Восточной Антарктиде здесь только вот эти дафнии. Есть немного, конечно, коловраток есть, олигохеты, и практически больше ничего нет.
Спасибо за
ответ. Явок нет, Андрей.
Thank you very much for your answering. I have one small question which arose in my mind in context of your answer. Вы упомянули, что они хорошо культивируются. У вас есть опыт культивирования этих дафний в Беларуси? Вы их привозите и культивируете или нет?
Мы сейчас, нет, я культивировал их в естественных условиях, брал воду, когда на самой станции. Вот. Я привез эфипиумы этих дафний. Мы сейчас сделали установку, которая и так как у нас есть гидрохимия этих озер, мы сейчас делаем гидрохимию, подгоним гидрохимию этой местной воды, то есть воды в этих аквариумах, и установим температуру в пределах 6-7 градусов для того, чтобы попробовать эфипиумы эти из эфипиума выклюнуть этих дафний. Но вообще это редко получается, как из литературы. Я знаю,
из литературы, да. То есть вообще-то массово их не культивируют, да?
Нет, их вообще никто не культивировал еще ни разу.
То есть вы просто попробуете это?
Да, это просто попробуем их культивировать. Но сейчас мы собрали большой материал и из различных озер на генетику этих дафний. И вот, может быть, с помощью наших дорогих украинских коллег, может, у нас что-нибудь и получится, так как эти озера все разные по типологии, по-разному по возрасту, и вот эта генетика нам может сказать, является ли в данном случае, может быть, вот это озеро Рейк центром, откуда расселение этих дафний проходит уже по всем другим антарктическим озерам. Так как 130 тысяч лет озеру это довольно-таки много. Ну, из таких еще мы исследовали, я хочу еще сказать, что мы сейчас, это не на белорусской станции это работает, а на белорусской станции мы сейчас взяли керн из озера 2 метра. То есть такой керн, 2-метровый керн, изъяли из него и немножко начинаем работать с ним по диатомии и по плесеням, так сказать, по этим грибам, которые сейчас на разных уровнях мы по горизонтально начинаем высевать это. Получается очень уникальные такие данные, я бы сказал, даже очень редкие такие. Нижняя граница этого керна – это около 20 тысяч лет. И все равно там происходит высев этих организмов. Такая интересная штука.
Да. Спасибо вам огромное за презентацию, за то, что вы взяли участие в нашей конференции. And I told thank you very much to Dr. Giginyak, and I invite the next speakers, Olena Bublik. Have we an Olena?
Yes, I am here. Hello.
Okay, hi, nice to meet you.
Could you please share my presentation, because I have problems here with sharing a full screen.
Okay. Sorry, moment.
Okay, dear colleagues, today I would like to speak about bioinformatic analysis of gene promoter of the Shamsia antarctica, DADREP2B stress-inducible transcription factor. Please, next slide. The Shamsia antarctica is one of the two vascular plant species that have undergone adaptation to survive in unfavorable conditions of low temperatures, high soil salinity, low humidity, etc., typical of the maritime Antarctic. We analyzed known regulatory cis elements in the gene promoter of DREP2B transcription factor, which is involved in response to drought, salinity, cold, and heat stresses. For the search for the 1000 base pair promoter sequence upstream of the DADREP2B gene was performed using the BLAST program in the GenBank SRA archive containing genomic DNA sequencing data of the Shamsia antarctica. For comparative analysis, we included 13 grass species from 3 subfamilies differing in cold and drought tolerance. The Shamsia antarctica, Gordium vulgare, Aegilops tauschii, Cicale cereale, Brachypodium distachyon from subfamily Poidae, Cetaria italica, Cetaria viridis, Cenchrus americanus, Zea mais and Sorghum bicolor from subfamily Panicoidae, and Oryza sativa, Oryza coarctata, and Oryza officinalis from subfamily Oryzaceae. Using the BLAST tool, the DREP2B promoter sequences were searched for in genomes of these species available in GenBank. Next slide, please. We studied the diversity of DREP2B gene promoter sequences for a sample of 13 grass species. Evolutionary distances by the method of maximum composite likelihood were 0.004 to 0.906, an average of 0.621. On the dendrogram constructed by the neighbor joining method, 3 clusters corresponding to subfamilies of the Poaceae family were formed. Analysis of regulatory elements in DREP2B promoters of 13 grass species performed using the plant care database revealed 54 cis-actin elements involved in response to abiotic and biotic stresses, light, and hormones, including abscisic acid, auxin, methyl jasmonate, ethylene, gibberellin, and salicylic acid, as well as tissue-specific cis elements. Among the found cis elements, the most abundant were those related to abiotic stress response that is consistent with the function of DREP2B transcription factors. Next slide, please. In the Shamsia antarctica, DADREP2B gene promoter, basal cis-actin elements CAD box and TATA box were found. Among those associated with abiotic stress, we found a number of hormone-sensitive cis-actin elements, namely sensitive to abscisic acid, auxin, methyl jasmonate, ethylene, gibberellin, and salicylic acid, as well as the binding site of MIP transcription factors and STRE, stress response element associated with the response to heat, osmotic, and nutrient stress. In the group of biotic stress-associated cis-actin elements, jasmonic acid and elicitor-responsive element GRE was found, which was absent in all other species. Light-responsive cis-actin elements included GATA motif and G-box. Tissue-specific cis-actin elements, CAD-box and D OCT, are both related to meristem expression. That DREP2B gene promoter didn't contain some abiotic stress-related cis-actin elements such as CAD, BOX, DRE, and MIC that are binding sites for transcription factors MIP-HV1, DRE, and MIC respectively, which were identified in DREP2B promoters of a number of other analyzed species. Next slide, please. Conclusions. Except for some individual variations, DREP2B promoters of the studied grass species, which have different cold and drought tolerance, and Antarctic extremophile, the Shamsia antarctica, were found to contain a similar set of cis-actin elements that suggests the similarity of the regulation of DREP2B expression and its potential function in response to stress in the studied species. This study of regulatory elements of JEB2B promoter in grass species contributes to the understanding of the regulation of this gene and forms the basis for further studies on the interactions among JEB2B and other transcription factors in stress-induced signaling pathways. Next slide, please. Thank you for your attention.
Thank you for very interesting presentation, Olena. Another one about Antarctic hairgrass adaptation mechanisms, and probably some questions.
Можно вопрос?
Конечно.
Да? Скажите, пожалуйста, экстремофил все-таки это растение, а почему его нет в Восточной Антарктиде? Что невозможность переноса какого-то?
Olena, one moment. Dr. Givignac asked about why this extremophile, the Shamsi Antarctica, absent in Eastern Antarctica.
I'm sorry, but I'm not the special. Может, мне стоит по-русски отвечать все-таки?
Я пойму.
Извините, я не специалист в этом вопросе. Я думаю, Ваня сможет вам именно экспертно это объяснить. Но я занимаюсь совсем другими вопросами, прошу прощения.
So Olena, you can repeat on English.
Okay, I can repeat that I'm not a specialist in this question, and unfortunately, I can't give this information. And I ask Ivan Parnikoza to help me if it's possible.
Thank you.
You are welcome, Olena. I consider that conditions in Eastern Antarctica over. Okay, in Russian at first. Условия в Восточной Антарктиде кажутся такими, которые как бы уже превосходят все возможности любых экстремофилов сосудистых растений. So, and in English, there are so extreme conditions, more than adaptive, all adaptive range of this, of course, extremophilic species, the Shamsi Antarctica, because Antarctic dry valleys or another continental sites very dry and very exposed to UV and another factors, so this is a little bit more than Antarctic plants can than conditions when they can survive. Yes, probably another questions.
Еще, пожалуйста, Иван, пожалуйста, если мы ее туда переселим, будет ли это являться инвазией?
This is Dr. Gigignac asked, is it possible to plant this the Shamsi Antarctica plants in the dry valleys on the Eastern Antarctica? Probably it will be invasion, but I consider that it is we need not to do this because we know exactly that no area for no possibility to exist for this vascular plants in this region. From the Antarctic Treaty System point of view, of course, is not probably very difficult question because probably Pete can ask can help me because transfer inside Antarctica not so strict regulate in Antarctic Treaty System. And in по-русски эти растения как бы там не выживут, поэтому как бы сенсу переносить их туда нельзя. Нет как бы на верную гибель. Но с точки зрения права как бы перенос растений из одной части Антарктики в другую в принципе не запрещен, но и сурово не зарегулирован. То есть поэтому как отнесутся к этому, что бы это было, это точно не будет инвазия, потому что растение просто не будет способно породить что-то, что может заселять дальше Антарктиду.
Спасибо. Спасибо.
another questions, comments? Okay.
Yeah, I do have one question, a quick one.
Okay, go ahead, Wong.
Yeah. Thank you, Elena, for your wonderful presentation. Just wondering, this is based on predictions. How do we test whether the prediction is correct? I mean, the promoters regions. Are you going to fuse it to a reporter gene and express it in this juncture, or how? Yeah. Thank you.
Mike.
I'm sorry, the sound is not very good. If I understood correctly the question, you asked about some genes that we also use for analysis of this features of this extremophile. Actually, we started from studies the genes of DREP2B transcription factors, and this next stage when we study its promoter. So we studied the group of DREP2 transcription factors and DREP1. All these transcription factors are stress inducible. So one feature interesting that we found here was about this gene DREP2B, about its alternative splicing forms. It has some interesting feature with its basic splicing form that normally is functionally inactive, but in the Shamsi Antarctica we found interesting point mutation that caused appearance of new start codon, and this makes this functionally inactive form to become functionally active, and this may be one reason that makes this species so possible to withstand these stressful conditions.
Okay, thank you.
More questions? Okay, if no, we all thank Elena Bublik for this additional green presentation. Yeah, and I invite the next speaker, Evgenia Prekrasna. Evgenia.
Hello. Do you hear me well?
Yes, go ahead.
Great. Oh, sorry. Okay, do you see my screen?
Yes, full screen, please.
Yeah, for sure. Okay, dear colleague, happy. What's wrong?
Something wrong. Please try to.
Maybe try again.
You don't see my screen, yeah? No.
No.
Okay. And now?
Yes.
Okay. My work is devoted to plant-bacteria interaction in Antarctica. So there, as you know, there are only two vascular plants in Antarctica, the Shamsi Antarctica and Colobanthus squitensis. You can see them on the slide. And these plants face really severe conditions in their native environment. That are low temperatures, soft freezing cycles, intense UV light, drought conditions. And on the other hand, endophytic bacteria, those bacteria that inhabit interior plant, part of the plant, are known for their plant growth promoting properties because they can help plants to survive some abiotic and biotic stress. protect them from parasites, enhance their nutrient acquisition, and so on. It's really intense direction in the agriculture and is used practically actually. So the question is, what is the role of endophytic bacteria in survival of Antarctic plants, whether they are helpful? So the first step of this ambitious direction is to know what is the composition of these endophytic bacterial communities of Antarctic vascular plants. So this work I'm sharing with you is devoted to this question. During 2018, in February, during the 24th Ukrainian Antarctic expedition, a number of samples were taken along the Western Antarctic Peninsula, mainly along the South Shetland Islands and in the close vicinity to the Argentina Islands. So there were samples of both Deschamps and Colobantus. They were taken aseptically and brought to the laboratory of the station of Vernadsky Antarctic Station. And there, the plants were surface sterilized by ethanol and sodium hypochlorite, just to get rid of the epiphytic bacteria and to work further only with endophytic bacteria that are inside of the plants. We worked only with the upper part of the plants this time, only with stems and with leaves. After the surface sterilization process, total DNA was immediately extracted there. And after that, those DNA was sequenced. The 16S RNA gene was sequenced on Illumina mice stack. And the further analysis was performed in CHIME2 and RStudio. Our sequencing output was really nice. As you see on the reflection curve, it shows that deeper sequencing would not result in higher reads estimates. But when we proceeded further with analysis, we saw that most of the sequences belong to the chloroplast of the plants. That's really a common situation in the 16S metagenomic sequencing, but when you work with plants, there are really a high number of chloroplasts. And when we filtered those data from our dataset, we considered that the OTU number, the number of operation taxonic units was really low in those dataset, and there was from 1 to 35 operational taxonomic units, and it was a really low number of them. So we dismissed the samples with the lowest number of OTUs for our further analysis. And actually, the first reason that was assumed for such low number of OTUs was that the quantity of DNA was not enough for sequencing enough of the bacterial 16S RNA reads. But when we analyzed the data, we saw that there was actually no difference in OTU number between the samples with higher DNA output and lower DNA output. And actually, there were some samples that had really high, comparatively high OTU numbers in the samples with low DNA content. In our further analysis, we saw that the Shannon index was predictably low for our samples. As you see here, for both the Deschamps Antarctica and Colobantus cretensis is about 3 on average, and comparatively to the other plant endophytic communities, I indicated here it on this part of the slide. This is the data for the plants that inhabit Atacama Desert in Chile. Those diversity indexes for those plants are quite higher. And actually, the other, our assumed reason of the low diversity was probably the rhizosphere, that is a source of endophytic bacteria, was not so bacterial rich. But actually, from our previous studies, we saw that rhizosphere microbial communities of the Deschamps Antarctica has really high estimates of bacteria. It has a really high diversity, so it's not the reason. So probably those plants suffer from some such abiotic stresses, and that is why bacterial colonization of the plant can be complicated due to the common plants cross tolerance, because interaction of different biotic and abiotic stresses, plants produce different peroxidic compounds that make colonization much harder for bacteria on the plant. The film composition of these Antarctic endophytic communities was that Proteobacteria, Bacteroides, and Actinobacteria were the most abundant phyla, and there were actually no just big difference in composition of communities between Deschamps Antarctica and Colobanthes cuitensis. Logically, the major bacterial classes of these bacteria were composed of proteobacterial classes alpha, beta, and gamma, with lower quantity of Syngobacteria and Actinobacteria, as well as bacilli. Some of the samples contained the minor classes representatives. They are shown here. On the family level, the most abundant families were Pseudomonaceae, Oxalobacteriaceae, and Sphingobacteriaceae. You can show them, see them here in this corner, while other bacterial families had rather patchy distribution, but Rhizobiotes, Xanthomonadaceae, Chitinophagaceae were quite abundant across the samples, and I want to say that most of these bacteria are assumed to be typical soil bacteria that shows the the colonization is mainly bacterial colonization of Deschamps Antarctica mainly occurs through the roots from the rhizosphere to the endosphere. And here are our conclusions. Actually, I want to say that the main conclusion is the low diversity of the endophytic bacterial communities of Deschamps Antarctica and Colobanthes cuitensis. And to be on the safe side, we are going to get deeper in this direction and to complement our studies with samples taken from the 2 more seasons from 2020 and 2021, just to be on a safe side and to have more statistical power for our research. Because if these bacterial communities are indeed have such low diversity and it is common for them, that means that this ecological niche is so sophisticated and really unique, and it's really distinct from other endophytic bacterial communities. So thank you for your attention, and your questions are welcomed.
Thank you, Evgenia. And we wait for your questions.
Hello, just a quick questions. Is there anybody ever cultured some of these endophytic bacteria from Deschamps, for instance?
Yeah, there are several studies that addressed culturing of the endophytic bacteria, and those researchers addressed their ability to solubilize some phosphate compounds or to produce some proteins that inhibit ice crystallization. And actually, our next step was to culture this bacteria as well. So we have the collection of the endophytic bacteria either, and we study their ability to promote Deschamps growth.
Okay, my next question is really regarding to the diversity that you found. How similar or different are they from other grasses in elsewhere, for instance, from the continent?
We haven't started studying this issue because I just consider that we have not so much data to compare it with other endophytic communities. So I think that after we receive some data from our more samples, we'll compare them with other grasses from the Deschamps and other grasses.
Okay, thank you very much. Yeah.
Other questions? We now have in chat comment from the Pete Conway about possibility to transfer some plants and other organisms from point of view of Antarctic treaty systems. Please, you can see in the chat. If no question in chat. Thank you, Evgenia, for another very green presentation.
You're welcome.
Yes, and I invite Maria Pavlovska to presentation.
Hello, can you see and can you hear me?
Yes.
Okay, I will try to share my screen now. Hope everything will go well. Okay. For some reason I can't share it, but I will try once more. Oh, yeah, here it is. Can you see it?
Yep.
Okay,
yes.
Great. Okay. Okay, we are moving from green presentations to blue presentations. I'm working with marine microbiology. Mainly, I'm also taking part in plant research, but this is like my primary topic. Therefore, I'm interested a lot in some technical things to make my research the most valid, the most reproducible. So now I will tell you a small technical note about the optimized protocol for DNA extraction from low abundance Antarctic bacteria plankton samples for increased yield. So why this is relevant? Because everybody thinks that, well, DNA extraction is a well-established thing. It's not something very tricky to do. But when you're working with environmental samples, especially with marine bacteria plankton samples, it often is challenging to extract the DNA from your samples from the bacteria plankton cells. As an example, I'm showing here the heat map. And in the lower part, you can see this MB, MBS, and some other letters. And these letters are just indicating the different extraction kits. These are all commercial kits. And you may see that even at the level of class, there are strong difference in the abundance of certain classes between different kits. So the red color is showing the higher abundance and the blue color is lower abundance. And this is the same actinobacteria, but when it's extracted with the different kits, the abundance data is really strikingly different. And if we go deeper to the genus level, these differences are even higher and even more pronounced. So the basic idea is that DNA extraction from low abundance samples is challenging. And it often leads to the skewed abundance data on rare taxa, and sometimes even on the taxa that are not so rare. But if we're talking about rare taxa, it's really being influenced by the factor of DNA extraction method. And as a result, you get the overall bacterial community structure, which is skewed compared to the real one. And rare bacterial taxa does not mean that this is something that is not relevant to your area of research. Sometimes you are interested in these tiny differences, so it really does matter. This is our small experiment design. We collected the bacterial plankton samples during 3 seasons, but this study was done particularly on the samples from February, March 2020. And this is the part of the study targeting the relationship between bacterial plankton and phytoplankton in marine ecosystem near Argentine Island archipelago, where our station is located. So then we did different treatments of the samples, trying to increase the yield of the DNA from them. First, we tried different homogenization steps. We tried grinding in liquid nitrogen and just grinding and homogenizing without the liquid nitrogen. Then we used different filter size, either 1 filter or half of the filter, and we used 3 different DNA extraction commercial kits available. Of course, you can also choose some different methods, but from our previous experience with other marine ecosystems, these kits really did work. Therefore, we wanted to choose from those methodologies and protocols that worked before for us. How we validated our results? We did the DNA extraction quality control. We estimated concentration and purity ratios with spectrophotometer, and then we tested it for amplification success. We just tried amplifying the quite small fragment of 16S V4 region of bacteria, because this is the fragment we are usually using for the taxonomic identification of our microbial communities. As a result, we showed that higher purity solid DNA isolation kit really outperformed the other kits, both in terms of concentration and in terms of the average purity ratios. You can see that there are two purity ratios here. These purity ratios, this 230 and 260 and 260 to 80, they are showing the presence of some proteins or phenols or salts that are really, really bad when you have DNA extraction and when you're trying to sequence the samples, because all these compounds prevent efficient amplification. And if they prevent efficient amplification, you get the skewed sequencing results. So ideally, these ratios should be around 1.82, both of them. And we also tested additional ethanol wash. We tried to remove these compounds that caused these bad purity ratios, but it didn't improve the results, so it couldn't influence the results. And also the grinding in liquid nitrogen didn't influence our DNA extraction success. So we can see here that DNA extraction with this high purity Canvax kit, in terms of concentration, it's not outperforming the rest of the kit so much. But when you look at the purity ratios, you see that Canvax high purity kit really is more helpful and more suitable for our samples, because both blood and tissue and power soil have poorer purity ratios. When we tested for amplification success, we realized that actually only Canvax kit resulted in the normal amplification. You can see some traces, some fragment being here after using the power soil Denise kit, but the amplification success is much, much worse. And in one of the treatments, it's not visible at all. And we think that the main reason of this is not the DNA concentration, but the presence of these organic substances that cause the low and poor purity ratios. When we tested for any significant influences of the factors that we tested in our DNA extraction procedures, we realized that the only statistically significant factor was the extraction kit used, and it was significant for DNA extraction and for both purity ratios. But when we are trying to get deeper, and when we tested this higher purity soil DNA isolation kit, that was the best one from the kits we used for our samples, we realized that also ethanol precipitation time influenced the DNA extraction success, and the filter size is also important. Therefore, based on our short technical assessment of the DNA extraction procedure, we can recommend the higher purity soil DNA isolation kit from Convex Biotech with 2-hour ethanol precipitation, and we recommend to use 1 full filter if possible for optimal DNA extraction from low abundance Antarctic bacteria plankton samples. You can also use different other procedures, but to my mind, one should always bear in mind that when you're starting to work with the different ecosystem, with the different samples, you should always test several approaches to understand what result you're actually getting. Thank you for your attention, and your questions are welcome.
Thank you, Maria, for so impressive talk. And I hope we will work more effectively on Vernadsky with our samples of bacterial plankton. And I wait for your questions.
Yeah, I do have a question. Yeah, wonderful talk. Yeah, we have been spending months also trying to optimize the conditions to extract DNA from the the soil bacteria. Yeah. So it's really important to, especially looking at environmental bacteria, to get a proper method to extraction so that it's not biased towards certain bacteria. So my question to you is that, are you using a mixture of bacteria during the extraction, or is just a pure culture?
It's a mixture of bacteria. We are actually collecting the bacteria on the special filters with 0.2 micrometer pore size, and then we are grinding these filters, and we are processing the whole filter with different bacterial cells on it.
Sure. So do you see a balance of it, gram-negative and gram-positive bacteria being represented?
Yeah, actually, we didn't test for this difference, but I know what you mean, that the difference can arise from the different cell structure, and therefore you have different chance of getting your DNA. Therefore, in this kit that we are using, there is also a special step that allows you to increase the temperature incubation step, both in terms of temperature and in terms of time. Specifically for working with the bacteria with the challenging cell wall structure.
we know that. Just to share a bit of information, how we did it is that we do the extraction using different kits, and just like what you did, you see, from soil. And then we do a PCR amplification of the 16S and doing a DGG, and then we do a DGG and see the profile, to see which methods will give you more banding, on the DGGE. So that will give you a rough idea of how representative are different methods. And sometimes it's worth using two different methods and then combine it. If you are looking at environmental, combine the DNA extraction into one and do the 16S amplification.
Yeah, that's perfect. Really, we also, by the way, we also tested not only the extraction method but the preservation method, because sometimes when you're working in the field, you don't have the chance to normally preserve your sample in ideal -80 conditions, and not always you also have some special storage solution at hand. Therefore, we just published a paper with my colleagues at the beginning of this year. If you are interested, I can just share it with you in the chat. I can just drop it in the chat. It tests the influence of different storage method on the microbial community structure. And we also made 16S sequencing to compare how the actual taxonomic structure is biased after the different preservation methods.
That's right.
It might be just interesting for you.
Yeah, wonderful. Thank you very much.
Yeah, thank you for your interest.
Thank you, Michael. Any other questions? So if no, thank you for blue presentation, and we will go to the presentation of our colleagues from Belarus. I invite Katerina Gribanova to presentation.
Yes, I'm here.
Please go ahead.
Dear colleagues, I would like to provide to your attention a report on the topic physiological and biochemical characteristics of yeasts isolated from various ecosystems of Eastern Antarctica. And I want to start with the well-known phrase that life is like riding a bicycle. In order to maintain balance, you must move. Likewise, modern science never stands still, and more new technologies are constantly emerging all over the world. Microbiology is a striking example of this, and every year we receive more and more data on microorganisms isolated from different parts of the globe. I present to your attention the results to some extent of studies of soil samples from Eastern Antarctica, namely samples from hypoliths and endoliths in the area of Молодежная station and in the area of the field base Mount Вечерня, and samples of hypoliths from Mac Robinson land. The purpose of this work was to isolate pure yeast cultures from soil samples from polar regions, to study their morphological, physiological, and biochemical characteristics. Isolation was carried out on 6 diagnostic media, and according to the results of microscopy, 20 cultures of yeast origin were isolated. And as you can see, the isolated cultures are quite diverse in shape and cell size. When considering the features of the morphology of the colonies of the isolated cultures, one can notice a rather large variety of pigmentation, since were isolated white, orange, green, and pink yeast cultures. The primary study of physiological and biochemical characteristics began with the study of the temperature range of growth of the isolated cultures, and theoretically one could expect the isolation of psychophilic microorganisms, but no strict psychophiles were found among the studied microorganisms. We can say that in general the analyzed cultures were able to grow in a very wide temperature range from 4 to 37 temperature, which allows us to consider them as facultative psychophiles. At the same time, the optimum for growth was around 18 degrees. When grown in liquid media, yeast causes turbidity from precipitate, rain, or film of a different nature, which indicates the ability of cells to unite into mycelial structures. Studies have shown that about half of the isolates are capable of forming films of various morphologies on the surface of liquid media. The other part of the isolates show the growth in the form of a ring at the liquid interface, and 2 isolates weren't characterized by growth in the liquid media. It turned out to be interesting that all 20 studied cultures of Antarctic yeasts have capsules. They were stained by 2 methods, the negative ink staining method and the Bourigins method, examples of the results of which you can see on the figure number 3. The responses of microorganisms to stressful influences can be considered as examples of adaptive capabilities. One of these is resistance to various concentrations of heavy metal in the medium. We used copper sulfate in concentration from 0.01 to 0.3%. But when the metal was added to the media, the expected high resistance wasn't found in the isolates. It was found that all isolates showed resistance to copper sulfate at the concentration of 0.01%, and only 3 cultures were characterized by weak growth at a metal concentration in the medium of 0.3%. The investigated isolates were also kept for different times under UV radiation from 5 to 40 minutes, and the presence of viable cells was observed. In the case, only 4 isolates were identified with high resistance to UV radiation. Next necessary step was to study the presence or absence of various enzymatic activities, taking into account the already known temperature range of cultural growth, I mean from 10 to 37 degrees. As you can see in the diagram, most of the studied isolates were characterized by the presence of lipolytic, amylolytic, DNA, urease activities, and the production of ethers. A much smaller number of isolates exhibited proteolytic, cellulolytic, and pectolytic activities, excreted organic acids, and formed starch-like compounds. Next, for research purposes, we carried out FTR spectroscopy with the support of the Norwegian University of Life Science. The resulting spectrum was divided into regions. For example, as you can see, the region from 700 to 1200. These regions previously established by other researchers and are considered generally accepted. As you can see, these are the regions of polysaccharides, proteins, and fatty acids. According to the results, the studied isolates differ mainly in the polysaccharide composition of the compounds they produce. The spectra of fatty acids regions and proteins regions are largely similar in all studied samples. The physiological state of yeasts was determined by the presence of storage carbohydrates, one of which is glycogen. According to the results obtained, 11 out of 20 studied cultures accumulated insignificant amount of glycogen. In 8 of 20, this indicator was average, and only one culture showed a high ability to accumulate glycogen in cells. Next, carcinogenic yeasts can be fortified with lipids, up to 70% by weight. The study of this property was carried out at various stages of culture growth, and it was revealed that most of the studied cultures, after a long period of incubation, passed through the accumulation of lipid droplets in the cells. And this accumulation can reach 2/3 of the cell volume or more. Next, in order to search for promising producers of glycolipid biosurfactants, screening of yeast synthesizing metabolites with emulsifying and wetting properties was carried out. The study of the emulsification index of the culture liquid showed that in 3 out of 20 samples, the index exceeds 50%. In 7 of them, it is in the range of 20 to 50%, and 10 cultures less than 20%. According to the literature, the higher the emulsification index, the higher the destructive activity of microorganisms. Next, when droplets of suspensions were applied to a hydrophobic surface, it was found that they wet the surface more significantly than water. It was found that the suspensions of 5 out of 20 studied cultures have the highest wetting abilities. And the last one, natural utilization of hydrocarbon mixtures is essential for biotechnology and ecology. And one of these sources of hydrocarbons is mineral motor oil. A study of the ability of yeast to break down the oil layer revealed 8 isolates with high destructive activity. Summing up, I would like to note that the isolates have significant potential for the further research, which allows us to expand our understanding of microorganisms in polar regions. Thank you for your attention.
Thank you, Katerina. Дякую. Some questions from our participants. I have question. Do you have in your condition, in conditional in East Antarctica, Belarusian station, or probably in surroundings, some melanized black yeasts?
If I understand right, you ask me if we have black yeast in our collection?
Yes, yes.
Yes, in this year, my supervisor bring me from the collection one black yeast. We think that it is the general exoshell, but I didn't work with this yeast yet because I don't. I have it only sometime. I don't work with it, but yes, we have it in our collection.
What is very interesting for us in what biotopes on rocks, on the surface of the ground. So equation is interested for us from ecological point of view. What cover of some of such yeasts in your conditions? Another questions? Okay. Тоді вітанки для Маміна. We will go to the next speaker. Larisa Bakhmutova, please go ahead.
Hello, dear colleagues. My name is Larisa Bakhmutova, and I'm working in National Antarctic Scientific Center of Ukraine. Let me introduce my presentation about socio-spatial isolation and the psychological state of Ukrainian Antarctic expeditioners participants. Well known that the long stay in extreme natural conditions and the socio-spatial isolation of a small group leads to physiological and psychological state changes of team members. This is characterized by psychophysiological and psycho-emotional instability such as reduced performance, grumps and depression, low activity. This might lead to self-isolation and formation of microgroups which affects both to health and personalities interaction in isolated group. As a result, we have transformation not only of human health, but also the psychophysiological state. That's why the monitoring of physiological and psychological indexes is the important part of assessing the condition of expeditioners in extreme conditions of Antarctica. The expeditioners' conditions were assessed for the psychophysiological indicators such as well-being, activity, mood, performance, health status, and social psychological indicators such as satisfaction with relations with colleagues, satisfactions with the environment and work performed, life satisfaction. According to the method of multifactorial research by KRILL personality factor evolutionary in the dynamics at the beginning, at the middle, and at the end of expeditions, we studied the indicators of emotional and strong wild psychological states of winterers by factors C, emotional stability, emotion stability, G, low normative behavior, higher normative behavior, factor A, factor I, rigidity, sensitivity, factor O, calm, anxiety, factor Q3, low self-control, high self-control. For statistical analysis, we used descriptive statistics, mean, standard deviation, one sample Kolmogorov-Smirnov test, and per sample t-test. On the basis of the express method of the scaled self-assessment, and we provide the monitoring of dynamics of expeditioners' psychological state during 1 year of work at the Antarctic station. A 1-year team consists of 12 members. The results of dynamics of the psychophysiological components are present in the table 1. On this picture, you can see all 5 self-assessment indicators of the psychophysiological component of the expeditionary psychological states rise significantly during the first 4 months, April to July, compared to the result obtained before the travel to the Antarctic station. It's well seen for self-assessment of well-being, but for mood, this tendency is low. Further, starting from August, the average self-assessment of the indicators is reaching the lowest values during the last 2 months of staying at the Antarctic station. The most significant decrease is for self-assessment of well-being, performance, and health if we compare it to the highest average date for the team. The next table 2 shows the dynamics of the social psychological indicators. The next picture shows parameters satisfaction with relationships with colleagues, at the beginning of the expedition increased insignificantly if we compare it to the start of expedition. But these indicators have lost during the 8th month, November, while for all other indicators are decreased to the end of expeditions to 11 or 12 months, February to March. For our opinion, this negative influence on the expeditioner's psychological state is due to long-term staying and working in isolation group. This indicator, satisfaction with the environment, unlike all other indicators, did not increase, but even decreased in the first months of begin at the Antarctic station. We think that this indicator determines the most sensitivity for stress of expeditioners, which a period for the first step adaptation when arrived to the station. The parameter satisfaction with work performance shows the greatest decrease of all other 9 indicators. This is naturally explained by the routine of the work in quite uniform conditions with limited choices of other activities. Moreover, the members of team have very limited choices of recreations. This reduced of psychophysiological resources of the expeditioners' members and the growing decrease to return home as fast as possible. The expeditioners' life satisfactions began to decrease substantially, not in August, like all other indicators, but the 2 months before, since June. This indicator is most sensitive for increased mental fatigue during the expeditions. The next table 3 shows the factors of emotional and strong-willed psychological state. As a result, it was found that Ukrainian winterers have higher 66.7% and medium 33.3% levels of emotional stability, which indicates a fairly higher endurance, efficiency, emotional maturity, realistic modes. Under the influence of socio-spatial isolation, spanning from the middle of the end of the winter year, more than 36.4% of the team has low normative behavior, which can manifest itself in the form of disorganization, irresponsibility, lack of principle, sabotage of team norms, and desire for self-isolation. At the end of winter, only 27% of the team has a high normative behavior. It's characterized by a high level of awareness in achieving goals and making important decisions, responsibility, compliance with norms and rules in accordance with all values. We conclude the stable and optimal level of the self-assist indicators of the psychophysiological component of the expeditioner's psychological state during the first third of expedition could be explained both the high-quality selection of members and mobilization of their mental and physical resources. Further decreasing of the indicators quite naturally occurs due to the gradual exhaustion of mental and physical resources and one-third of the polar night and the influence of other negative natural factors which affects of people, life and work at the Antarctic station. This data can help in the development selection criteria and physiological support of groups of specialists working in conditions of long-term isolation, such as members of polar expeditions, oil platform team, and crew of long-distance ship, submarines, and space missions. This topic is very interesting and important for us. Why we have prospects for future researchers in this year. 12 people take part in 26 Ukrainian Antarctic expeditions, and we provide the monitoring of the physiological and psychological state as well. Thank you for attention.
Thank you for very interesting presentation, Larissa. I hope we all understand that we need to pay a lot of attention to changing of the crew members for Antarctic expeditions, and probably some questions.
Thank you.
Participants? No question in chat. Okay, thank you. And I can invite Natalia Raksha with group for presentation. Are you here?
Yeah.
Yes. Okay, go ahead.
Share my presentation, one moment. Oh no, it's not my presentation. Oh, sorry, I see some problems.
Do you need my assistance? I have your presentation.
I tried, one moment. Okay. Okay, I suppose you see my presentation, yes? It's okay?
Yes.
Okay.
Full screen, please.
Yeah. Full screen?
Full screen, please.
F5. Yes. Sorry.
You can press button on the top of your window.
I know, function 5.
No, I mean button, I mean by mouse. You saw button near save.
Кнопочка внизу.
Я вижу, но что-то...
Или внизу, или вверху, наоборот, самого вверху, видите, там, где дискетка, рядом стрелочки, и потом это,
Можно
на
так оставить?
оранжевом фоне.
А так можно оставить? Это так некрасиво.
Там кнопочка такая внизу, не класснуть.
А я это понимаю, но я не понимаю, почему я не хочу...
Вот близко, так. Вот.
Ну, конечно, я переключилась на другой этот монитор.
Okay.
Ну, окей. Ну да, у меня теперь два монитора. One moment, maybe I can... Може ви підключите, тому що у мене підключено два одночасно этих, и она друг другу мешает.
Так, давайте.
Давайте тогда вы, потому что так не получится. Sorry. Окей. Окей. Можно начинать, да?
Так, вперед.
Dear colleagues, let me introduce my work titled Antioxidant peptide derived from the Antarctic hydrobiomes. Free radicals are fundamental to living organisms and represent an essential part of metabolism, but an imbalance between the formation of free radicals and their elimination potentially leads to damage of cells and development of some serious pathologies associated with oxidative and nitrosative stress. Therefore, the reduction of the level of free radicals is probably one of the most effective ways to protect cells against oxidative damage. In this context, antioxidant peptides drive significant research attention. First of all, due to their numerous beneficial health effects. They are safe, they are quite stable and easy absorbed, and what is very important, their low molecular weight excludes the development of immune response. And marine organisms tend to be promising material for peptide production. And in recent years, special attention is paid for hydrobionts from the Antarctic region, which are poorly exploited and potentially can be a valuable source of new biological compounds. In our previous study, we found that peptides obtained by enzymatic hydrolysis of tissue of Antarctic hydrobionts and Adenosine colbecki exert protective effect on activity of some antioxidant enzymes, and they also decrease the level of lipid peroxidation, and they preserve the proteinogenesis and non-proteinogenesis of sulfhydryl group. Next slide, please. So in our current study, we are focused on isolate peptides from the tissue of Antarctic hydrobionts, such as Antarctic krill, Antarctic jellyfish, and Antarctic limpet, and analyze their biological activities. To obtain our main goal, we determine some tasks. First of all, it needs to optimize methodological approach for obtaining natural peptide with molecular weight up to 5 kilodaltons from the tissue of Antarctic hydrobionts. Next, to study antioxidant activity of peptides derived from the Antarctic hydrobionts and investigate their membrane protective activities, and to evaluate the effect of peptides on key factors of blood clotting. Next slide, please. Next slide. No, previous one. Yes, thank you. The common approach to obtain bioactive peptides include acidic or maybe alkalic hydrolysis, as well as enzymatic hydrolysis using appropriate enzyme. But in current work, we focused on isolation of naturally occurring peptides. And peptide fractions from the tissue of Antarctic hydrobionts were isolated by sequential precipitation with perchloric acid and ethanol. And the next obtained supernatant was lyophilized. And picture number 1, we see the conformation of purity of obtained peptide fraction. And you see that molecular weight of isolated peptide fractions are within a range of 1 kilodaltons to maybe 6, maybe 7 kilodaltons. And next, to obtain more information about peptides, first of all, their structure, we performed Wade electrophoresis. And this approach is very interesting because that approach allows separating moleculars not only by their molecular weights, but also in accordance with their isoelectric points. And at figure number 2, you see result of Wade electrophoretic analysis obtained peptide fractions. And you see that isoelectric points, peptides that were isolated from tissue of Antarctic hydrobionts located at alkaline range. It means that dominant part of peptide represented by cationic peptides. Next slide, please. Considering the complexity of oxidative process taking place in biological system, as well as different mechanisms by which compounds may realize their activity, using a single approach to evaluate the antioxidant potential of compounds is not suitable way. And in our current study, various types of assay were performed to estimate the antioxidant activity of peptides derived from the tissues of Antarctic hydrobionts. And according to obtained results, all hydrobionts peptides have an antioxidant activity, but their effectiveness was different for different species. For example, peptides isolated from Antarctic krill are effective in scavenging diphenyl picosyl radicals and also in scavenging superoxide anion radicals. You see it my slide, and also in scavenging nitric oxide radicals. Also, peptides derived from Antarctic krill have higher reducing activity compared with peptides from Antarctic limpet or Antarctic jellyfish. Antarctic jellyfish peptides were very effective in scavenging hydrogen peroxide, and also they possess metal halating activity. And Antarctic limpet peptides were effective in scavenging of hydroxyl radicals. Next slide, please. Next, we evaluate the membrane protective property of peptides. And even though peptides from Antarctic krill were more effective in scavenging free radicals, these peptides didn't show any the ability to protect erythrocytes from hydrogen-induced hemolysis of erythrocytes. And according to obtained data, only peptides derived from Antarctic limpet had a slight protective effect against chemically induced hemolysis of erythrocytes. And you see that this peptide reduced the level of erythrocyte hemolysis by 27% compared with control, for example. And next slide. And then next stage, the effect of peptides derived from tissue of Antarctic hydrobionts on Boris polymerization of fibrinogen and activity of thrombin were investigated to evaluate their antithrombotic activity. And at first, the influence of peptides on the ability of fibrinogen to polymerize was investigated. As can be seen from picture number 4, pre-incubation of fibrinogen with peptide derived from the tissue of Antarctic hydrobiont before the addition of thrombin didn't affect the polymerization process, because both dynamic of this process as well as value of extinctions were within control level. Taking into account that thrombin is an ideal target for pharmacological correction, we analyze the ability of peptide to influence the amylolytic activity of thrombin and activity of this enzyme against fibrinogen. And according to obtained data, the polymerization of fibrinogen was increased or enhanced after the addition of thrombin that was pre-incubated for 15 minutes with peptide derived from Antarctic hydrobionts. And the effect was similar for all peptide fraction. The level of fibrinogen polymerization was increased, you can see from this picture, comparing with control. Next slide, please. At the final stage of our work, the effect of peptides on the amylolytic activity of thrombin was analyzed. And it was found that peptides of Antarctic jellyfish as well as peptides of Antarctic krill didn't exert any significant influence or effect on the amylolytic activity of thrombin. In contrast, the pre-incubation of thrombin with peptide derived from Antarctic limpet led to decrease the amylolytic activity of thrombin. You see this control, and you see the results of influence of peptides on amylolytic activity of thrombin. We see then limpet peptides were peptides from Antarctic hydrobiont, Antarctic limpet were more effective comparing with this one. And summarizing, the tissue of Antarctic hydrobionts have naturally peptides. They possess different types of biological activity, and they can be used as antioxidants and also can be used as molecules that affect blood coagulation. Thank you very much for your attention.
Thank you very much for your presentation, Natalia, and about bioprospecting from on the base of marine organisms from in Antarctica. Probably some questions. In chat, no question in chat. Okay, thank you, and okay, thank you for your attention. See you.
Что означает 3 p.m.?
Few records of the southern right whales movement between warm water breeding grounds and the Southern Ocean as far south as 65th parallel south have been documented. In 20th century, there were only a few records of right whales in Antarctic waters. In April 1986, 8 southern right whales were sighted in the Gerler Strait. It was the southern limit of known distribution for the species, 64th parallel south in comparison with 63rd known from previous records. However, in some later studies, no sightings of southern right whales were recorded for the West Antarctic Peninsula area. In some Antarctic and Antarctic waters, southern right whale's prey is Antarctic krill and copypods, and the West Antarctic Peninsula continental shelf is important for krill stocks. It is the northernmost part of the Antarctica and biologically productive area experiencing marine ecosystem transformation caused by one of the fastest rates of climate change on Earth. The purpose of the present study was to reveal whether southern right whales are present in the waters of the West Antarctic Peninsula in austral summer. Vessel surveys were conducted in 2018, 19, and 2020 in the waters of the West Antarctic Peninsula. Penola Strait and the French Passage were main research locations. 81 research cruises were conducted in different seasons using both sailing yachts and research and fishery vessels. The total length of the cruises was 1,673 nautical miles. Visual observations were conducted under good or moderate weather conditions. Cetaceans were detected with the naked eye, and binoculars were also used for observations. The following data were collected: GPS tracks and observational effort, weather conditions, group size, composition, and behavior. To confirm species identification and for the individual photo identification, images were taken using professional digital single-lens reflex cameras with telephoto zoom lenses. Photo identification has been used to recognize and recite individual whales. Unique patterns of head callosities and skin pigmentations were traditionally used. The best photographs of the identified whales were added to the initiated photo identification catalog of the species and shared with other researchers. 2 sightings of the southern right whales occurred during our studies. On April 7, 2018, first specimen was encountered. There were several groups of humpback whales in the area between the northeastern part of the Hovgar Island and Kiev Peninsula of the Antarctic Peninsula, and 1 adult southern right whale was sighted in a group with 4 adult humpback whales. Second encounter of the single southern right whale happened on April 21, 2020 in Gerler Strait near the southeastern coast of the Brabant Island. In 2018, observations were performed from Zodiac boat in the middle of the day during 1 hour and 15 minutes from a distance of several dozen meters and more. Intensive social interactions happened between southern right whales and humpback whales, swimming together in a close distance, different patterns of surface movement, synchronous dives, intense loud breathing, and pulse. Most of the interactions happened between southern right whale and 1 humpback whale in particular. And by the end of the observations, they were separated from 2 other humpback whales. After the separation, their behavior changed to slow travel side by side. Southern right whale and humpback whale interspecies interactions with elements of possible sexual behavior were previously described with the southern right whale breeding ground of Mozambique. In 2020, a southern right whale was noticed from the vessel Betanzos. Single individual was traveling. The photographic images of the southern right whale encountered in 2018 potentially could be used for the identification of this specimen. Images were shared with some holders of the photo identification catalogs of the species in the Southwest Atlantic, but no match were found yet. Sightings of the southern right whales never happened during previous 22 Ukrainian Antarctic expeditions based at academic Venatsky Station. However, cetacean observations until 2018 were rather opportunistic than dedicated. Results of our study indicate the autumn presence of some southern right whales in the West Antarctic Peninsula on the southern edge of the known range of the species. It is likely that whales were taking advantage of changing climate conditions and used ice-free areas during the austral summer to move far south to feed on krill. As a part of long-term year-round cetacean research, regular visual coast-based monitoring and passive acoustic monitoring should be also performed to increase observation effort and to better understand seasonal distribution and density dynamics of cetaceans in the area, which we plan to perform during our 26th Ukrainian Antarctic expedition. Recently, we managed to collect information on some other sightings of the southern red whales in the waters of Antarctic Peninsula, provided by colleagues from other countries, and I initiated writing of the collaborative research note. At the moment, we don't have any information of more southern sightings of the southern red whale than our sighting of 2018. However, according to the personal communications of Dr. Ari Friedlander, our colleagues from Palmer Station encountered one female in Limer Channel, which is quite close to our sighting location. I would like to express my gratitude for the field, for the help and research to employees of the National Antarctic Scientific Center and to members of the Ukrainian Antarctic expeditions, crew of sailing yachts and vessels, and to other people involved. Thank you for your attention. I would like to share with conference participants very warm wishes from members of the 26th Ukrainian Antarctic expedition.
Dear Oksana, thank you very much for your presentation. I invite our participants to look at this nice photo in which you can see our winterers, which are right now at the station. And please, maybe somebody has questions. Have we any questions or not? Okay. Unfortunately, we haven't questions, Oksana.
Possibly not question, but little comment. Now we took part in the historical moment, because that is the first conference talk directly from Ukrainian Vernadsky base. I hope that is the first direct participation of Ukrainian wintering team in international conferences, but not the last.
The last, yeah, I hope too. Yes, it's a very big event for Ukrainian Antarctic research. And yes, I hope in nearest future it will be possible for many another peoples to participate, not only Ukrainian Antarctic expedition, conference, maybe in another events, including the seminar, which will be in Malaysia. And I ask all researchers from our Antarctic base to think about submit their report for this seminar too. Okay, thank you very much, Oksana. Can we back
Thank you
to
very
our...
much for this opportunity.
Okay.
Can I ask a question?
Yes, of course, you can put question. I invited everybody.
Yes.
Put question, please.
So, Oksana, are you doing anything dealing with the ocean productivity, things like chlorophyll A, or looking at ocean colors and whatever, just to have a link of the oceanography to where you locate the southern right whales?
Yes, we collected phytoplankton samples, environmental DNA samples, and water samples. And yeah, did some hydrochemical analysis as well. So yeah, if I understand you correctly, you will try
Are you doing any current analysis too?
Yes, so we collect biopsy samples from whales and collect water DNA samples and do.
No, what I mean is current, the current, the flow of the ocean. The current.
Current, no. It's the special task for our meteorological group. So they monitor it regularly. Mm-hmm.
Okay.
Right now it's a station, so they do it.
Okay, thank you.
There is a question in chat box. There is a question
please.
from Tatiana Kuzmina.
Okay. Oksana, can you see chat box, or maybe I should read for you?
Yeah. Yes, question about to extend area of observations. So it's very tempting to have this possibility to extend the area, but my goal is to implement like basic monitoring at Vernadsky station, to make it work. So it's only third year of dedicated cetacean research, and I believe it's very important to make it more comprehensive now. And so now we try to include acoustic observations in our monitoring program. So maybe for now it's the best thing to do it to stay near Vernadsky and to work on this program, multi-year research. And lately, maybe it will be nice to extend the area of our research.
Thank you. Okay.
Yeah, are you interested to put a hydrophone in for passive monitoring?
Yes, so we have 2 hydrophones this year. One will be deployed as a passive acoustic monitoring buoy, and another is portative. So we use it from boat, from the shore, like, yeah, when whales are around. So we combine both methods.
Yeah, we do something quite similar with dolphins, where we listen. We have one PhD student who was doing that in observing dolphin, but it is more in the South China Sea than
It's very interesting.
Yeah, but passive sampling.
Sorry, our colleagues who work in our center at the same times investigate dolphins of Black Sea. Yevhen, maybe you tell some about this investigation? No? Okay, sorry. Okay, sorry. If you will interesting, I will give you contact and you will be able to discuss with them more detail about dolphin investigation.
Yeah, we are monitoring the Irrawaddy dolphin, which is quite a rare species actually in the Bay of Brunei. Thank you.
And in the Black Sea, we use seaport detectors to detect harbor porpoise and 2 dolphin species, common dolphin and bottlenose dolphin. So yeah, it's first year of monitoring as well.
Yeah, we have bottlenose too in our South China Sea, but the one that we are more interested in is the Irrawaddy because it's more, it's on the IUCN list, if I'm not mistaken. We have quite still have quite a number still in the South China Sea, our side. The northern side is a bit difficult because of overfishing.
Yeah, it's very interesting.
Yeah. Okay, it seems to me, Oksana, stay with us and continue our discussions because maybe no new idea. Because it seems to me your presentation very nice flow to the discussion of our future possible collaboration between Ukraine and Malaysia. And that is why I would like to say that we made the first step of this, and right now